red blood cell, white. Hi, welcome to CSEC Biology, the cover page. Let's talk about components of the blood. Can you identify the components of the blood? Let's try. Red blood cell, white blood cell, platelets, plasma, red blood cell, we were correct. Phagocyte, that's a type of white blood cell. Lymphocyte, that's also a type of white blood cell. Platelets, and of course, plasma. Red blood cells are also called erythrocytes. They are made in the red bone marrow. Approximately 5 million is found in a millimeter cube of blood. This increases the surface area of the blood to move gases quickly. Red blood cells are said to be biconcave discs, which means that there is a depression on both sides of the cell, and this increases the rate of diffusion for the blood cell. They transport oxygen, which combines with hemoglobin to make oxyhemoglobin. Red blood cell also carries some amount of carbon dioxide. They are the most numerous component of the blood since the plasma is not counted. They have no nuclei. They are short-lived, about 120 days, and are destroyed by the liver and spleen. Here we look at the diagram of a red blood cell, and this area shows the concave area of the cell, and it is on both sides of a red blood cell, which makes it 2 bi, hence biconcave. White blood cells are called leukocytes. The white blood cells serve as part of the body's defense mechanism. They are made in bone marrow, spleen, and lymph nodes. They are not as numerous as a red blood cell, as only approximately 10,000 is found in a millimeter cube of blood. They are, however, larger than the red blood cell. There we have it, the phagocyte, a type of white blood cell. The lymphocyte, another type of white blood cell, and we must be reminded that these are not the only types of white blood cells, but of course, you'll continue the research. Lymphocytes, they make antibodies that destroys pathogen. They have a central nucleus. It is important to note that we are not going to be covering other types of white blood cell for this lesson. However, you'll be treated with information on the phagocytes and, of course, lymphocytes. Phagocytes engulf foreign organisms through phagocytosis. They have lobed nucleus and are usually granular in structure. They are usually irregular shape. So if you were supposed to look at the phagocyte here, you'll realize the lobe nucleus. Here we have a lobe nucleus, and it seems to be somewhat, it has a rough coat, somewhat granular. That's what it means here. It has this rough coat and the lobe nucleus. And of course, it would pretty much put the body in the shape of the lobe nucleus in order to carry out phagocytosis. The lymphocyte is somewhat different. Thrombocytes are platelets. Platelets, they have no nucleus, as they are just small fragments from the red blood cell. They are irregular in shape and are usually found in fragment groups. So usually the platelets are pretty much in the blood like a group here, and they might not be represented like this on your exams, but usually once we're looking at blood cells and we're seeing these small fragments together like we're seeing here, it pretty much tells us that that's the platelet. They are colorless, and that's important to note. So if we look at what should be the blood vessel here, we would see the different components. Now this is really simplified, as you are seeing this yellow area which represents the plasma. Really, it doesn't appear to be like that in the body, but for you to identify the components, we would have simplified and made all these pretty visible for you. Platelets are much smaller than red and white blood cells. Only approximately 250,000 can be found in a millimeter cube of blood. Platelets are important in clot formation at the site of a wound. Plasma. Plasma is a yellow part of the blood that is made of approximately 90% water and the rest made of solutes. And that's going to be about 10%. It is the main transport mechanism of the blood. Plasma supply water to the tissues and of course all cells. It carries hormones from gland to target site. Plasma carries nitrogenous waste to excretory organs. Plasma also transports nutrients from the intestine to liver and tissues. It helps to transport heat in the blood. Plasma also transports carbon dioxide as bicarbonate to the lungs. Plasma carries all the components of the blood, and hence it makes up 55% of the blood. Products from plasma are used to treat patients with burn, trauma, and other emergencies. It's important to note that the red blood cell lacks a nucleus, as it increases the space of the red blood cell to carry the added oxygen needed for respiration. Now tell me, can you identify the cells on the screen? Can you identify the components of the blood? Tell us in the comments below. Thanks much for watching, and please be reminded to like, share, and subscribe. And when you do, click that notification bell and select all. Also be reminded to join us on a Wednesday, Friday, and Sunday at 5:05 p.m. for live classes from Kingston, Jamaica. Thanks for watching. Now, All right, great. We're fine now. All right, good evening and welcome to the show. I am Mr. Wilson, and we are streaming from Kingston, Jamaica, in the Caribbean. The emphasis here is to assist students from across the Caribbean in acing the biology and human and social biology exams. We have been we have been working with the students for a year now, and we are pretty much pretty close to the end of the journey. I like to identify persons on the show. There are some persons who have always been with me. I am seeing Macmillan, who is always here with me, and there are a number of persons. I can't call all the names. I can remember Tanisha. I can remember Amoy. I can remember Otisha. Those persons were like among the very first persons who shared the platform with us. But since then, we've had many other persons who have joined and continue to be with us, continue to trust the brand, and continue to support us in every little way that they can. That is pretty much getting us the content that we can prepare for you. I observed in the chat when I came in earlier that persons are saying that sir need to put in a little HSB in it. I understand the anxiety that persons really need to see the topic HSB on the paper. Today is Saturday, but because I have you in mind, I must say I went to my workplace today and I fetched myself papers that I'm going to be working for HSB. These papers are going to be out. At least one will be out as of tomorrow. So we're going to be having two shows tomorrow, one for biology. Well, it's two shows for everyone. I continue to say to students that if you are studying biology or HSB, it is very important for you to know both syllabus if you can, if you're allowed to expose to them, to be exposed to them. Because what I've observed from working the past papers and teaching both subjects, I'm seeing in some cases the content on the bio papers, it is as if something that was lifted from HSB and vice versa. And in some cases, it's the same questions on both papers. But you probably wouldn't know because you're not paying attention to both papers. But I am paying attention to both papers. So when I say that, you understand. For example, this is the HSB 2018 paper. And if you look at the paper, you are seeing respiration there. I could pull a lot of bio paper that I have in front of me now, and they have respiration just the same. So to say that, look at the menstrual cycle. How many bio paper you have seen so far with the menstrual cycle? Or with the female reproductive organ and the menstrual cycle as well. How many of these have we seen? So yes, the syllabus, they are a little different, but there are a lot of things that are common. It is as if when it comes to biology and HSB, it is as if you are making that distinction between plant cell and animal cell. Yes, there are a few things that are different, but for the greater part, they are just cells, and a lot of things are pretty much similar. Welcome to the show. We had a great time this morning. I didn't even know that we spent so much time online. We spent 4 hours online. I was up from 2 o'clock because I was really, really excited to meet the new folks and to ensure that we're all prepared for classes for the exam come tomorrow. I fell asleep today, but I am back well charged and prepared to share with you on the 2018 paper. That's the paper that we're going to be looking at tonight. Now, you would know by now that I am not the teacher who is going to come and unload what I don't have in my head to you. I'm here to upload, so I'm cheating you. So I want to know what is it that you have to share, and then I will assist where I can. There are too many persons in the room for me to name persons now, but I observe I'm seeing Trinidad here, so I want to big up Trinidad. I'm seeing Guyana here. I want to big up Guyana. I know that the Jamaicans, they are here. And if you just want to say which country you're from, let's just do that, and then we are going to hop right into the lesson. We started on the show with a video that looks at the components of a blood. I gave you that video because we're going to need that information for a question in the paper. And sometime throughout the presentation, I'm going to go back to video from my channel so as to assist you in answering questions. I know that a lot of persons would not have been exposed to things like mitosis, meiosis, and I keep doing the genetic question. We have done 6 past papers, paper 2 so far, and there is one question that is an all 6 papers, and that is the genetic question. What we've observed in all 6 papers, if they are not testing for sex-linked inheritance, well, it's sex-linked inheritance, that's the one we've been seeing. Color blindness and hemophilia, that's a common one. So we know that we have to prepare you. The other thing that we have found common in all 6 papers that we have looked at between 2016 and 2021, almost all these papers have a graph question. Interestingly tonight, while we spoke about using the first column as the independent variable, when we look at the 2018 paper, there is some anomaly as to the shape of the graph. So I'm seeing a lot of love coming in, Turks and Caicos. I can't even call that country name, but it's a wonderful place, I was told. Welcome to the show. Great. So I looked at the paper and I was going through the paper today and I realized that there was some anomaly with the graph and how we have presented graph so far. Now, CXC gave us a table on this paper, and you know I am the advocate to say do the graph question because it's easy. So I'm going to just show you this graph question. That is how this paper begins, and from there you can assist me in solving the graph. Now, of course, remember when you get to the exam, you are going to read everything on the paper. So this is the May, June 2018. And I said tomorrow I'm going to be giving you the 2021. And we're not going to be having a class tomorrow because we want to put out some paper tomorrow. All right, here's the question. And I hope you can see it well on the screen. So we are ready to go now. I hope that everybody is in the house. So I'm going to just increase the font size here. And then we can go right ahead into this question, and I'll be asking the question, so be armed with the answer. So here we are required to answer all questions in this section. Now, scientists interested in studying the survival of organisms living at high temperatures identify some microorganisms living in a desert habitat where the average daily temperature exceeds 50 degrees Celsius. I am seeing some clue here, so I want to start looking at the clues. It exceed. There must be some reason why they are using this exceed, right? Now, they extract an enzyme. So it's an enzyme here that they extracted, and it is a protease. So it is pretty much acting on protein, that's a substrate, that facilitates the digestion of protein. So protein being the substrate here, from these organisms, and investigated the effect of temperature on the action of enzymes. So here we are going to be investigating the effect of temperature on the action of enzymes. Now, I hope that I read it well for you. If not, I hope that you are able to see it and you are able to read it for yourselves. However, you are tasked with the responsibility of stating 2 precautions that they should take in carrying out this experiment. For me, when I looked at the question, there are some things that popped out. And this is one of the things that popped out for me. Microorganism. Microorganism, it said a lot for me in this precaution. It is as if you were going to a compost heap to pull some millipedes or something. What are the precautions that you definitely will be taking? And remember, precaution are things that you're going to be, steps that you're going to be taking in order to ensure that you are safe. So tell me in the chat, what are 2 precautions that you'll be taking in ensuring that all goes well with the lab, nobody is injured, everybody will be fine? What are the precautions that you'll be taking? All right, great. If you are just joining, I am Mr. Wilson from CSEC Biology TCP. We have close to 300 videos on the channel. I know that you'll be doing exams on Monday and you're definitely going to pass. So we are expecting that you're going to pass the information to students of law school so that they will benefit come next year. Somebody wrote me about the SBA for HSB. If you are sitting the exams this year, then the HSB is not yours. However, if you are sitting the exam next year, that might somewhat differ. CXC spoke to that in a couple of articles, but to be honest with you, I can't remember. So you want to make sure that you are wearing a gloves, because a gloves is pretty much a protective equipment or a protective. All right, so I'm just going to show some on the screen. All right, you want to wear a gloves. You want to wear. Somebody saying that you should wear their coats. Persons are saying that you should wear gloves. Let me see what's happening here. I'm sharing it on the screen. Uh, be careful while extracting the microorganism. Handle it with care. Anybody who would have heard about all the rumors around COVID would have, of course, remember that you want to handle this microorganism with care because you really don't know much about the microorganism. So these are some of the precautions that you're definitely going to take. Handle that microorganism with care, wear appropriate PPE. All right, so I guess everybody would know PPE. And let me see what's happening there again. Wear the necessary gears like gloves. All right, and we're coming with a lot of things here. Let me see what else. You want to wear your goggles. That's a safety equipment. So I'm getting a better feeling as to what teachers are telling students. Be gentle so as not to hurt the organism. We're talking about microorganism. I don't know how gentle we can be not to hurt them. I really don't know. I'm very sorry. Ensure the microorganism only in stay in the similar pH and temperature. So definitely, because we are looking at the enzyme there, we want to ensure that the enzyme, we know that enzymes are easily denatured. So you really don't want to expose this enzyme to too much of a low temperature. That's a precaution you want to look at. You really don't want to expose this enzyme to too much of a high temperature. Neither do you want to expose this enzyme to arsenic. So you want to make sure that you're taking those precautions. You want to ensure that the enzyme is not exposed to too much of an acidic environment. Yes, I know that it's a protease, but you want to ensure that it is kept in the appropriate medium. So these are our precautions that you want to take. Now, the fact that we don't know this organism and it is a microorganism, then we could easily pretty much what's the word? Well, a school of thought is that there's a possibility that this microorganism could be pathogenic. We don't know if the organism is pathogenic. So I'm giving you a whole lot of precautions here. We don't know if the organism is pathogenic. So we really want to treat that organism with care, and we really don't want any spillage in an area that the organism really doesn't belong. So we must handle and treat those petri dish with care, making sure that there's no spill. And of course, we won't have contamination of that microorganism in the area that we are working because pretty much we know but little about the organism. Ensure that equipment are properly cleaned. So you want to ensure that equipment are properly clean for this particular experiment so as to ensure that the experiment will not be contaminated. I think I like the answers that I'm getting. Ensure that the microorganism remain at the right temperature because temperature could definitely denature the organism if it's not at the appropriate temperature. So we want to pay attention to that. Very good. I think we did well there. You need to give yourself a round of applause. We're going to be moving down. Most person would have scored 2 marks because there are so many precautions to be taken or to be addressed here. So of course, it would have been easy for many persons to get the mark. Now, here's where we're going to be having a little challenge. Now, the results of their experiments are presented in the table. On the grid provided in figure 1 on page 5, draw a graph to represent the result presented. So you're going to get 4 marks for that. Now, the first thing we need to think about to score that first mark is that the graph needs to have a title. Now, the title that we're going to be using for the graph, it's going to be the title from the table. All we're going to be doing is to remove table here and put a graph. So what we would have observed with all 6 papers that we've looked at so far, or probably 5 of the 6 papers, you would have realized that it's either we're using information from a table to construct a graph or using information from a graph to construct a table. But it's the first time that we're seeing this type of a table that we're going to be using to construct a graph. Now, one might ask, why is the table looking like this? Now, that is easy. The table is looking like this because CXC wants to find out if you understand the difference between independent variable and the responding variable. That's what CXC is testing here. If you did your planning and design, can you really determine what is the independent variable versus what is the manipulated variable? Now, the independent variable, one will recall, goes on the x-axis, and the responding variable, did I say, I should be saying independent and the responding variable. The responding variable, that is going to go on the y-axis. So what I want you to tell me here, what are we going to put on the x-axis here? We have temperature and we have time taken for the protein to disappear. So tell me pretty much in the chat, what is it that we're going to be putting on the x-axis? Because we're going to need to know that in order to construct the graph. So let's go right ahead. We are looking at the Biology 2018 May-June paper. What are we going to put on the x-axis? Wow. Yeah, it's an independent variable. But what is the, oh, so the independent variable is temperature and the dependent variable is time. All right, so Philip, I remember this morning, there are some things that just stand out for me. This morning you said that you felt like you were going to fail. Now, what I want you to do for me, Philip, your answer is correct. So I'm not going to be going all over the place. What I want you to do for me, I want you to explain to your classmates now, what did you use to determine the independent variable versus the responding variable? I want you to do that for me. And if you are not certain, class, what's the difference between independent and dependent variable, I invite you on going onto YouTube and look at the difference there. You really don't want to go to the exam and just think that time is always it or that is always it. You want to make sure that you are using empirical data to address. If you observe here, a lot of persons are saying time because we're accustomed to seeing time, and some literature might say time. But when you get to understand the difference between independent variable and the dependent variable, you will understand. Now here, you will observe that what we are manipulating here is pretty much the temperature. What we are manipulating here is pretty much the temperature. What we are manipulating here is the temperature. And it's the temperature that becomes the independent variable, that which you are manipulating. I was expecting Philip to come back with some answer, but Philip is not yet. So remember, that which you are manipulating, we're going to get it on the x-axis, and I invite you to continue your studies on independent and dependent variable. That should have been a skill that we should have mastered from we completed planning and design. So Philip is back with us. Let me share on the screen what he says. I am going to be reading it for you. Okay, for the independent variable is the things that is being manipulated, all right, which will affect the time taken. So basically, what happens on the independent? So here you observe that we are manipulating temperature. Welcome to the show. If it's the first time for you here, we're manipulating temperature. Now, there's something that is going to stick out here, the fact that this graph tends to be a little different than the graph that we are accustomed to. So we are now going to be plotting this graph on the grid here. I do hope that you have a paper. I tend to like to go the shortcut way because we have a time challenge. So the first thing we want to do, let us start doing the thing right. So the graph needs to get a title. So you're going to write your title on your paper. So you need to have a title for your graph. And the title that we're going to be using will come from the title in the table here. So we're going to use this bit here as the title for the graph, just like we're going to remove the table. The next thing that we want to do, we know that temperature is the independent variable here, So we are going to be having temperature on the x-axis. Great, so those are labeled. So we are going to come on down, and what we want to do is not just to populate the grid here. It would be nice if you use your ruler to pretty much draw the line around. Now, having done that, and I'm teaching you to do the graph just more than answering the question, the other thing we need to have in is our directional line. So it is increasing in that direction. Then we also need to name the axis. Now, naming the axis does not mean that we are only going to leave it at the y-axis. Now, the name of this axis, the name of the y-axis, you're going to get it from the table here. So this is going to be the name of the y-axis. That's what you're going to write on the y-axis. Now, the same is true for the x-axis. So we are going to be, now your graph would have had a title already. Here's your title for the graph. Didn't see that. Directional line, and again, we are going to name this axis. So this axis, the name of this axis is going to be temperature, and the unit is degrees Celsius. So you're going to write it in nicely there. All right, beautiful. So now that we have done that, it is time to populate or to determine a scale. So look at the numbers here. We really need to find a scale. So if you look at temperature, it is increasing in increment of 5 in some places. So we have 5, 10, there's no 15, we have 30, 45, and all of that. So what we know here is that we have 1, 2, 3, 4, 5, 6, 7, 8 bit of stuff to place on the graph. Now, could we increase in increments of 5? You are going to do this at home. I'm just going to be touching the thing. So we have 1 there. So we could probably start here with 5. And then this would have been 10. And you can do the maths. 10, 15, 20, 25, 30, 35, 40, 45, 50, 55, 60, 65, and we stop here at 70. I do hope that you have this past paper and you're following. We stop there at 70. All right, great. And then on this side, we could increase in pretty much similar increment. Let us look at what is happening there. So we just did for temperature all this information. Now, if you look at the time taken for the protein to disappear, meaning that the protein would have been digested, the time taken, we are looking at it here. Now, we are going up to 60 just the same. All right, with the lowest figure here being 15. Now, could we go up at 5 increments of 5? I'm not too sure. One thing that we want to do is to ensure or try as best as possible to complete at least 75% of the graph. So if we were supposed to start here, I do hope that it will work. 5, 10. Oh no, we don't want to start there. Let's let me clear that. We could start with 5, but that is going to it's going to cause the graph to be a little too crowded. We don't want the graph to be too crowded. Though we want to get 75%, we want to use the area efficiently. All right, so here we go. So here we could start at 10, and then of course this is going to be 20, this is going to be 30, this is going to be 40, here we are going to have 50, here we are going to have 60, here we are going to have 70. So we could just write that 70 in, right? So we could increase by 5 or we could increase by 10. And now we have that awesome responsibility of plotting the curve. I do hope that everyone is following. Is everybody with us? Sir, we can use 2 centimeters to 10 units. All right, great. Yes, yes, that's cool. All right. What works for you? Pretty much just try that. Let me just get this off the screen. I'm very sorry. I hope that you would have learned from what Philip shared with us. Thank you very much, Philip. We always like when students share. All right, the only reason I am working on the graph because I fully don't understand how to get you to work on it that everybody can see on this platform. All right, so we go back to the graph. I think I need to make it a little bigger that for those persons who haven't mastered graphs as yet. All right, so we know that that is 70. So this is naturally going to be 60, and this is going to be 50. And remember that you have to write in the names. I hope that you are writing the stuff in as I speak and not only just looking like you're watching a movie. You want to ensure that you are practicing the tactile skill. You want to get that in. So we are going to be plotting first. So we are going to plot from X to Y. So it's going to be 5 degrees Celsius to 60. So let's go on down and look at what is happening here. So 5 degrees Celsius. We are going to map that onto 60. So wherever it intercepts, that's where we're going to plot. We're going to plot right there. This is where it intercepts, right here. And we could use a cross. Remember, if we are using a dot, we are supposed to draw a circle around it. So right where it intercepts there, and then I'm almost certain it's going to be pretty much something similar. It's going to be 10 to 60 and 30 to 45. So 10 to 60, right here, 10 to 60, and 30, 5, 10, 15, 20, 25. I think this is 30. You can solve it on your paper just the same. All right, so it's 30 to 45. Let me see how that goes. So it's going to be 10, 20, 30, 40. So 45 is somewhere there. So we are going to be solving it there. So let us see how we could get that intersection. All right, so we're going up with 30. I'm just going to mark this line that I can find it as soon as I move up. So we are supposed to be intercepting here, right at that spot there. And then we are going to now go again, 30 to 45 that was. We are going to go to 45 to 25. So 45 to 25. So if this is 30, then naturally this is going to be 30, 35, 40. Then this is going to be 45. So we are going 45 to what? I hope you're paying attention. Those persons who don't understand. So we are going 45 to 25. Sorry. 45 to 25. So this is 45. We're going 10, 20, 25. So we are going to be right here for 25. And then I keep going up and down. I do hope that you are looking and you could just write it down and just do it. All right. Then we're going to go 15 to 8, 50 to 18, and then 55 to 15. Wow. So 55, so 45, 50. So we're going to have 55 here. I think we said 55 is going up to 18. So this is going to be 10, 15, 16, 17, somewhere here. 18. That's what it was. 55 to 18 and then 50 to 16. Oh, 50 to 18 and 55 to 15. So 55 to 15. All right. And then 50 to 18. All right, so great. We need to remove this. All right, great. And then we move on up. Going back. And then we have 60 to 60 and 65 to 60. So we're going to go up 60 to 60. So 50. So we're going up the smartest line here. We're not going to be using that. It's just a marker for me to remember. So 60 to 60. We go here. All right, so I think I can erase this now. Erase this line, and I'm supposed to erase this bit right here as well. And then we are going to go 65 to 60 just the same, if I remember well. So 65 to 60, there we go. And 65 to 60. All right, 60 to 60 and 65 to 60. Now we're supposed to swing this curve. And of course, I am using, I'm using my touchpad on the computer. So let me see how best I can do this. So if you look here, we have a constant here. So it's a straight line there. And then we are going to swing on down. Make sure that your line is coming down as accurate as possible. Right down. And then we're going to curve it there. And then we are going to go back up. And if you observe how sharp that is, and then we are going to go that way. Tell me if that's the shape of your graph. All right, so let me see what you have there in the chat. So I'm back with you in the chat. Is that the same graph that you got? All right, so I am looking at you now. So tell me if your graph is looking like that. Before we proceed, is there something you think I need to change? Is there something that we need to explain? Okay, so Washington, your graph is looking like that. Beautiful. Guys, if you're talking to me, I'm a little old, so I really don't, laugh. I don't know what these three-letter stuff means. I don't even know if it's IRM. I'm very sorry. Just assist me. You should not use a ruler to draw that curve. All right, just try to swing your hand like I did to draw the curve. It's only at where you have the 60 and you have that constant, you probably could do that. And if you observe, when I came down to the bottom, I didn't make a V. I made a smooth curve because it connotates something if you make a V. It means that you would have stopped and started. But that is not what happened. There was a change and it went through a gradual curve and then it went up. All right, great. A lot of persons are assisting me here and I am most grateful. All right. We're looking at a 2018 biology paper. You probably could find it online somewhere. That's the paper we're working out now. So while we have this discussion, it is expected that you are going to be writing on your paper so that you will have your own stuff. All right, great. Smooth curve. So we can move right along now, right? Great. Is everybody fine? Everybody's fine? Anyone have that paper can send it to me, please. All right, if there's anybody who knows where the past papers are, I know past paper is all over the place. You can probably post a link here. Is that an optimum pH graph? I am going to take this from Casanova. Let me look at this. Casanova is asking that question. Is that an optimum pH graph? Usually, it is the different way around. Thank you very much. It is the first I am seeing it like that. Can you explain, sir? I am in the same position that you are in. I'm in the same position that you are in. But the reason why it is like this, it is as a result of where the independent variable falls. But what you could do for us, this is a science class, you could put the content of the y-axis on the x-axis and draw the graph and tell us what it looks like. All right, you are allowed to do that. And if you observe, each time I ask, are you with me? Because we're working as a team. And I continue to say, feel free to say, no, sir, let's do it another way. All right, great. So we are, I don't know if anybody else would have seen this before. I've never seen it like this before, but it makes a really interesting question now that we're going to be moving on to this question that now you are supposed to answer this. This is what, is it Philip was asking, Casanova was asking about. So here, you are going to tell us for the graph in figure 1, identify the optimum temperature for the protease activity. So the optimum temperature, you are going to write it in the chat for me. Optimum temperature. Optimum temperature. So there's the graph. Probably I need to give you a full shot. What's the optimum temperature? Let me come on over and take a look there. What's the optimum temperature? If you observe, Casanova wrote something on the screen there, and this particular question, it is not the question that we're accustomed to. Somebody said 65 temperature. Oh, that's a question. 60 degree. That's a question. 60 degree, 60 degree. Ah, 60 degree. What went wrong or what went right? 60 degree is the optimum temperature. What is the optimum temperature? Guys, what's the optimum temperature? All right, I see some persons coming with some other answers. So we have 60 so far, we have 65, we have 55. Remember, it is very important for you to write whether it is right or wrong. Now, when it is wrong, we get an opportunity to correct. And when it is right, we get an opportunity to question you as to how you came up with that answer. So I am assuming that many persons would have entered their answer. So now I'm going to take you back to the table to show you how you could shortcut this method. Now, the reason why some persons are getting it incorrect is because it's how the brain works. The brain is accustomed to see the graph the other way around. So if we were, just let me go to the graph and show you something. If we were supposed to go the other way around and we had to use our ruler to come straight down here, then everybody would be able to tell me that the optimum there is 55. Everybody would be able to tell me that the optimum temperature is that. Now, it's the same concept that you are going to apply. The same concept you are going to apply. But look here. One might say that it is not true. Let us go back to the table. Did you observe this arrow that I placed here? Now, let us look at the shortest time for digestion. The shortest time for digestion is going to be the optimum that the enzyme is working. Because remember, the whole idea of chemical digestion with the enzyme is to get the food broken down, and you want this to be done very quickly. And that is why we have mechanical digestion before chemical digestion, so that we can get this process moving on very quickly. So if you observe here, at 55 degrees Celsius, it takes only 15 minutes to break down the protein. At 50 degrees Celsius, it's taking 18 minutes. So the first thing we want to identify, what is our objective here? The objective is to determine how long, what temperature will affect the enzyme, and at what temperature the enzyme will work best. Now, the objective of the enzyme working best here is to digest, and it said it right up here, to digest the protein. So if the protein is being digested in 15 minutes, the quickest time of all the times here, then we are almost certain that the optimum temperature is going to be 55. So it's right there in the table. And if you understand tables, you could have gotten that information from the table without even drawing the graph. So remember, we missed out one thing on the graph that we did just now. I am not even going to quiz you on what we missed out. I'll just talk about it so we can move on. Remember, we missed out on the scale. Always remember that you want to draw in your scale on the table. On the graph, you want to do that. So the graph and the title, please follow the best practices that I would have assisted you with here. All right, great. I think most persons are comfortable. Let me see what you're saying over here. Okay, great. I left out the scale. Wow, I love that. All right, so let me just get this off the screen and then we're going to move on into the other question. And what you're going to find here is that the questions that we are looking at on this particular paper, they are somewhat similar to the questions on the 2019 paper and I think 2016. I do hope that my memory served me well. All right, so let us look at some of these questions and you're going to answer them for me. So you want to make sure that you are looking at your graph because you're going to need that information to answer the questions. So here we go. First question, and I'm going to make it really big for you. The first question to be answered here, we're supposed to suggest one reason for the shape of a graph at less than 10 degrees Celsius. Now, when you see something like this on an exam paper, simply put, it is simply asking you about the properties of enzymes. That is what it is asking you about. So it is saying that, let me just read it again. So just one reason for the shape of the graph at less than 10 degrees Celsius. Two reasons for the shape of the graph between 10 and 55 degrees Celsius, and one reason for the shape of the graph above or more than 55 degrees Celsius. So I'm going to come right back up and I'm going to show you where they want that information from. So just remind me here, 10 to 55 and 55. All right. So I'm going to change this here, change my ink. So they want to find out about the shape of the graph right here. Great. They also want to find out about the shape of the graph from there, coming down here. So that is like A, they want to find out the shape of the graph there. Like B, they want to find out about the shape of the graph here. And they also want to find out about the shape of the graph there at C. So if you want to say ABC, you can. So let us just up on back. You could just draw that very quickly on a paper because I'm going to move it from here. Just sketch the graph very quickly on a paper and they want to find out about the graph. Now here we go back to something that we have been working on some time now. When we said that, just let me draw these. We use these, fluctuation, constant, or that should have been constant, increase, decrease. Remember we looked at those, all right, for curves on a graph, how to interpret them. Now, what would have caused things to be pretty constant? All right, when things are constant, there's like no change. It is like every holiday you are getting the same rows. When it is Teacher's Day, teachers continue only to get a glass. It's a cup, a hand teacup, right? So that is like a constant. Every Teacher's Day, some teachers know that they are not going to get anything. That's a gift for the teacher, or they are going to get a cup, one of those stuff. All right, so we want to answer now. You want to explain for me the shape of a graph? You are watching CSEC Biology TCP. I am Mr. Wilson. This is, of course, our YouTube channel, but we have an online school, and it is, I think it's not on screen. It is TCP Academy. You could just type that in and you will find us. You can register for classes for September for biology and HSB for now. Others will be added in short order. If you want to sponsor any of our program or promote an item on the channel, you can use that email address to reach us. All right, great. So let me look at what is happening here now. All right, the enzyme are inactive. All right, let me just show some things on the screen for you. We want to see from everybody. All right, buffering. Okay, it's buffering. All right, let me look at Gobbin. All right, so Gobbin answer is on screen. Let's look at that. The enzymes are inactive at temperatures below 10 degrees Celsius. But Gobbin, let us look at what is happening here. This is not below 10 degrees, you know. This is between 10 and, let me look at it. This is between, is it below 10? It is below, oh yes, it's between 5 and 10. Okay, I'm sorry, Gobbin. All right, great. All right, so the enzymes are pretty much inactive below 10 degrees Celsius. So what that means is that there's pretty much no activity there. All right, remember, enzymes are not organisms, so they can be denatured, but it can be denatured but not killed. All right, let me see what's happening with anybody else who would have said, okay, so we have A answered. So what about B? The drop at C is because it is denatured. But what had caused it to denature? All right. So the shape of the graph at C is a result of the enzyme being denatured and whatever. So you give us that explanation. But what would have caused the enzyme to be denatured there? All right. So let me look at, I'm seeing something a while ago. It just flipped away from me. Yeah, which would mean, okay, let me put back this on screen. So which would mean the temperature is too high, I think, for the enzyme to work effectively. That sounds good if you ask me. All right, so the temperature is too high, so the enzyme was denatured. So we are saying at A, the temperature is too low. We are saying at C, the temperature is too high. All right, great. So let us look at something else. I want to hear from everyone. Oh, at 10 degrees to, let me put this one on the screen. At 10 degree to 55 degrees, as temperature increased, there was an increase in the I got you on this one. Philip. Philip, please go back to the curves on the graph and look at the direction of that curve. And that is why we have directional line on the graph so that we don't make that mistake. Philip, please revisit that content. Look at the curves on the graph. We have fluctuation, we have constant, we have increase, and we have decrease. So look, try, remember that time is on the y-axis. Is it that time is increasing or decreasing? Pay attention to that, please, everyone. Look at the little arrow there. Those are the things that I am looking for just to make you stronger. Okay. Lol. Okay. As the temperature increased, but super Cody, I don't know what you're talking about. I don't know if you're talking about A, B, C, or D. So that is not helping us much because we are not going to assume. We want to ensure that you're saying what you want to say, that if we are supposed to make corrections, we can make them. All right? As the temperature increases, the enzyme activity increase. All right? So let me look at this one. So look at this one, Bowen. As the temperature increases, the enzyme activity increase. While we said in our last class, if it's going up diagonal, it's exponential. If it's coming down like this, it's also exponential. So this is a pretty nice answer for B. I didn't get to look at everybody's answer, but once we have the right answer, I do hope that your answer is within line. Thanks for the participation. As we move to the other question, your answer might be the answer coming on screen next time around. So I think that we were pretty, we think I did a pretty fair job with those questions. But look at this, look at this. This question, this particular question, now I remember, it is a repeat of the 2016 paper. Describe a suitable procedure that the scientists could use to investigate the effect of pH on the activity of the protease enzyme. So now you are supposed to write a procedure. So writing the procedure, you have something already. How will the scientists modify that procedure? Or what would you do if you are supposed to test here for the effects of pH? Let me see in the comment. Let me see what you would do. Write it for me. It's your exam. What would you do? All right, so I am waiting on those comments to move on. What would you do? So now we want to look at pH instead of temperature. What would be required of us to get that done? I am Mr. Wilson from TCP Academy. And if we're on YouTube, it's CSEC Biology TCP or CSEC Biology, the cover page. We would have pretty close to 300 videos on the channel. So please make sure that you take part, and many are past papers on the channel, and we continue to load. We're very, very happy to be with you. We do hope that everyone will be doing well for the exams. All right, great. I did not say, but please be reminded, I have my books coming out for August. So they are going to be on all major platforms. So I'd love for you to go on out and have a read of a copy. Good night and thanks for joining. All right, let us see what will happen here. Can't think anything. Okay. Let me look at this. I don't know what it says, but I'm going to put it on screen. Use varying pH. That's a good way to start on the activity of the protease enzyme. Ensure that the temperature is held at constant. Use pH meter to test the activity of the protease. To test the activity of the protease. That part is a little challenging for me, Casanova, using the pH meter to test the activity of the protease. What are you hoping to get when you test the activity of the protease? Are you suggesting that the protease is going to change the pH of the content? I'm not too sure what you're going to get from that, from using the pH meter to test the activity. If you can share that with us, I can't really see through it. I'm very sorry. But if you can share it or defend your point, that would be good. Use different enzymes. Oh, no, we have to use the same enzyme. All we're supposed to do is to deal with the pH. It's pretty much the same enzyme we're using. All right. Place like 3 samples of the enzyme. Why are you making it hard for yourself? All you need to do is to change the enzyme, to change the pH. So stop using the temperature, as Casanova said here, and you are now going to be using pH. Place the enzyme in several pH level and observe the response. I understand what you are saying, Bowen, but you could have been a little more specific. What if you had said that you're going to be using 5 test tube with 5 different pH or 3 test tube? I think that would have been a little more accurate and would pretty much understand clearly where you are going. Now, I love, I want to see the setup, but something is missing here. All right, so let me see what's happening here. Place the enzyme, okay. Oh, I don't want a certain amount of time. Give me a time. If it doesn't work, just give me a time. All right, we are testing the protease enzyme, though we work in an acidic condition, I think. Okay, yes, it works in an acidic condition, but because it works in an acidic condition, you want to have varying pH, right? Remember that this organism is an organism that we're not familiar with. So a whole lot of things could have been different for this organism. So probably you want to look at acid and base when you're looking at that pH. Okay, sir. So instead of using temperature, the scientists can use different level pH to determine which pH the enzyme work best. All right. And in determining the different pH, that is where we are going to be using the pH meter. So now I'm going to be asking this question. Who put this on the screen? That was Philip. You said that? Okay, great. So I am going to be asking somebody now. I'm going to ask the room. How will we know? What are some of the parameters that what parameter can we use to determine the effect or the effectiveness of the enzyme at whatever pH? Can you tell me there? How will we know what pH the enzyme is working best at? Using the same enzyme and constant temperature. That is good. You want to make sure that the temperature is constant. Is that Ron? And use 4 samples of the same enzyme. And yes, that is also good. I like the specificity. And use gradual enzyme and use a gradual change in pH level and monitor the change in the enzyme. I think this is like one of the closest thing to being a procedure. I think I like it. All right. Maybe, just let me share it on the screen. All right. Let me say maybe. Oh, no, we're not going to go with maybe. We're not going to go with maybe. Just say what you have to say. Say it like you're writing an hypothesis. Varying pH condition instead of Cody, if you were the person before, all that I wanted to know is that I want you to understand that what the pH meter does is to pretty much check the pH or check for the hydrogen, whatever it is there. Unless you are using like a multimeter probe, like the YSMI multimeter probe, that probably could check for something else, but what it would check for, I don't know. Now, what I am looking for from persons, you would have done an enzyme lab somewhere, I see an animation somewhere. Now, when you place this this enzyme in the acidic environment, it is going to cause some effervescence, or one might say froth, or it might cause bubbles, but you are going to see some reaction. And based, what we usually do is to measure the height in the tube, and that will tell us where the reaction is greatest. Or you could count the number of bubbles, and that will tell you where the reaction is greatest. All right, great. You did well there. We spent a lot of time on that question, and we're still on question number 1. But the good thing about this paper is not all the questions that will be answered, because some of the questions will not be on the exam as far as we know. But this morning you said that excretion would not be there, but I am not in agreement with you. So we're going to be looking at the skin. This is a very easy diagram to label. The human skin. Just let me ensure that we're doing what we should do. Okay, great. So we're going to be looking at the human skin. And remember, this morning when we were looking at the 2017 paper, the skin was on the 2017 paper as well. The human skin shown in figure 2 is an organ which helps human to regulate their body temperature. Beautiful. Now, you are required to name the parts labeled 1, 2, 3, and 4. So I'm coming over to the chat to see what you have there. 1, 2, 3, and 4. The nice part of it is that all of us have a little skin. So you could just look at your skin and probably ask the skin, what is your name? Remind me, please. I need to write it in. Try something, but I want to know the parts labeled 1, 2, 3, so on and so forth. I want to know what name you would give to them. And it's very important that you know the names because we are going to be using these shortly thereafter to do something else. So let me see what you have. Okay, so one person is out of the pack saying the ear. Somebody is saying number 2 is the sebaceous gland. Interesting. All right. Somebody is saying that it is the sweat gland. I don't know what is the sweat gland. Oh, 2 is the sweat gland. Okay, so one person thinks that it is the sebaceous gland. One person thinks that it is the sweat gland. Another person thinks that number 1 is the ear. Another person thinks that 3 is the erectopili. I think that is beyond our CXC. All right. Sir, please zoom. I think we need to zoom. And I think we need to be a little careful with Google because we might just Google outside of the syllabus. It's nice to have new information anyhow. All right, so there we have it. It's a little bigger. All right, great. So let us come back over there. Some persons might correct what they had. All right, so 4 is the capillary, I believe. I see. And 2 is the sweat gland. Interesting. 3 is the ear erector muscle. So let me just pop on some things on screen so that we can get some answers. So I'm just randomly going to pull somebody. Oh, I want to pull a wrong answer first. I like wrong answers. A. What? Oh, ear follicle, which it is. All right. So it is pretty much an ear. And you can tell me if there's a difference or what's the difference between ear and ear follicle. Tell me what's the difference between ear and ear follicle, or if there's a difference between ear and ear follicle. Don't guess now. All right, so number 2 is, of course, the erector muscle. And of course, number 3 is, did I say number 2? No, number 2 is wrong. That is for the one that I'm reading there. So let me just try to find something else. I'm sorry. So let's go with that on screen, 1 there. And let's go with this, the answers there, 1. And let's go with 3. All right, great. That's 3, answer on screen. And let's go with, I saw somebody with the answer there. Let's let me look for the answer. I like you guys. It seems as if we are all ready for the exam. I am looking for the other answer. I'm looking for 4. All right, so I saw this earlier. So blood vessel or capillary, that's fine. Beautiful. We did awesome. That was awesome. We did really well there just now. You need to give yourselves a round of applause. So we're going to move on into the other question now. And we're still looking at the skin. So we scored full marks just now, full marks. All right, and we did that together. That was the collaborative effort. All right, so we're looking at the other question. Look at this question. This one is pretty beautiful. This was a question we were reading this morning on the 2017 paper. And this question has found its way on the 2018 paper as well. It said, describe all the structures named in BI will help human to regulate their body temperature. So I will remind you of what we named there. We named the air, we named the blood capillary, we named the sweat gland, and the air erector muscle. Now you have to tell me, how will these help an individual to regulate his or her body temperature? All right, so we are looking for words that fall under, what are they again? Vasoconstriction and vasodilation. So use those 4 words. Tell me what's going to happen during vasodilation. Tell me what is going to happen during vasoconstriction, and you are using those 4 words to defend your point. The sweat glands transport urea to the surface. But Gabin, I'm not too sure what you're answering there, but you are answering for excretion. But what we are doing now is to look at temperature regulation. So Gabin, we have to make sure that we are answering the right thing. All right, sweat is secreted into the, okay, you're answering Jita, you're answering for excretion. However, this question is a temperature regulation question. Like we know from our studies that mammals and bird, remember these 2 big words from biology, picilotherm and homotherm. So mammals and birds, they are homotherms. Sorry, they are homotherms, which mean that they are warm-blooded. They regulate their body temperature from inside. All the other organisms are picilotherm. They regulate their body temperature from outside. So we want to know, as a homotherm, you and I, how do we regulate our body temperature using these 4 listed, 4 parts listed, the 4 parts of the skin listed. So the ear erect under cold condition, but lies flat under hot condition. All right, let me read this. I don't know if there's a limitation as to what you can write, but the ear erect under cold condition, but lies flat under hot condition in order to keep the body temperature constant. So now you leave us in I don't know, they say that's a story hanger or something like that. What the ear up or down, lying flat, how does that help to regulate their body temperature? I don't understand. If you could provide some more, provide some clarity on that, I'd be really, really happy, because I don't want persons to leave here with a part of the information. I want everybody to be armed with the information leaving here. Now, when the temperature is high, the capillary causes vasodilation, causing the sweat gland to release sweat to cool down the skin. All right, so the sweat is going to remove heat from the body, and as soon as it gets to the surface of the skin, the air is going to move it, and then you're going to feel that cooling effect as the heat is moved. Now, as it were, for vasodilation on the blood vessel, the blood vessels are going to dilate. They are coming very close to the skin so that they can get rid of as much heat as is possible. Now, as it were, just let me up and back up here to something that I asked about earlier. So with the ear being erect on the skin, what happens is that it traps air from the environment and pretty much insulate the skin, thus keeping it warm. However, during hot condition, the air will lie on the skin so that air can flow over it easily, keeping you cool. Let me see what else is said here. So we have looked at the ear, and we have looked at the blood vessel, and we have also looked at the sweat gland. But what about the erector muscle? What is the function of the erector muscle in this all? The ear relaxes to allow air to, okay, circulate above the skin. So Gabin is back, and Gabin is being very strong. All right, let me see Kingsley here. The capillary, we're going to use the word dilate, not enlarge. We're going to use the capillary dilate. That's a term that we use with the blood vessel. It's either going to dilate or constrict. Enlarge means that it dilates just the same, but we just want to use the right terminology. To allow more blood to flow near the skin when the temperature is high, so blood can carry away the heat. Opposite for the temperature too cold. That is wonderful. So all those girls who are going to the bathroom half an hour before school dismisses, and you go to the bathroom and you spend half an hour to wash your face, and then you get back out of the bathroom 5 minutes before school dismisses, and your face look really, really cool. What you have just done is to cool down your face so much that the blood vessels would have constrict so much that you are not having this flush look on your face, this red look on your face. Instead, it is all cool like cucumber. So it's pretty much a little bit of biology that you went into the bathroom to do. So the next time you happen to get caught in the bathroom, just tell them that you went to the bathroom for a biology class. All right. The sweat gland produces sweat, which, let me just put it on the screen. All right, there. So what about the ear erector muscle? That's the one that I'm looking for now. What it does. So I've heard about the sweat gland. I have heard about the ear. I have heard about the blood vessel. What about the ear erector muscle? What does that? What does that? What's the function of that? All right, I'm seeing some big one coming here. Stimulate, dilate. I am. All right. So remember, in vasodilation, that is where we are cooling down the body, and vasoconstriction, that is where we want the body to become warm. And remember, this is for the bird would have done something similar, but not the exact thing. But like things like crocodiles and so on that are cold-blooded. Now, cold-blooded does not mean that you are wicked. That is not what it means. What it means. All right. So I did say earlier that the homotherms, or the warm-blooded organism, are mammals and bird. All the others, fish, reptile, amphibian, they are all cold-blooded. And they regulate their body temperature using external means. All right, so the ear erector muscle, I think I'm seeing it now. Let me see what it says here. That keep loading me. All right, here we go. What's happening? Every time I click on that, it goes to something else. All right, let me try this one down here. And then it's happening again. Oh, ear erector muscle controls the orientation of the ear. So the ear coming up, it's going to be the ear erector muscle. Going down, it's going to be. So the ear is pretty much controlled by that muscle. Thank you very much. We were really waiting for that. All right, we're moving on to the other question. You did well there. The collaborative effort, that was really, really wonderful. Now we're still at question number 1, you know. Explain how changes in the epidermal, this is one that might bowl a lot of persons. But let me see where we're going to go with it. Explain how changes in the epidermal cell of leaves help plant to conserve water during hot temperature. This particular question was on the 2017 paper we answered this morning. So tell me that. Let me see. Remember, on the 2017 paper, the farmer had this plant that wilted during the day and was flushed during the night. It's pretty much the same question. Same concept. All right, so let me see what will happen, how we are going to treat with that question. Let me put it on the screen. All right, so right after this question, I hope the HSB students realize that the enzyme question is a HSB topic. So after that, we're going to be doing more biology and HSB together, where we're looking at some of these stuff that are really related to biology. I do hope that the HSB students, they are taking note and understanding that enzymes, it's not an isolated topic to HSB. It's both bio and HSB. Great, great. Homostasis, okay? Great. All right. So I see Jetta coming in nicely there. Once you would have said the guard cell, pretty much you know you're armed with the information to answer this question. So let me just show it on the screen and read what you have there. Jetta came out of the block very, very fast. I want to put what you have on the screen. Okay, great. There we are. All right. So the guard cell close the stomata, preventing water loss at high temperature, reducing water loss through transpiration. But remember, let me just check the question before I say what I have to say. Explain how changes in the epidermal cell of a leaf help plants to conserve water during hot temperature. Okay. Is there anything else we know about the guard cell as to how the guard cell operates? Is there anything else we know about the guard cell? Now, for those persons who don't know where the guard cell is going, you would have realized that the guard cell is something from photosynthesis. You would have known that, right? The guard cell is something from photosynthesis. But more could be said for this 3 marks question than that which Jita said. You could have told me about the cell becoming turgid. When the cell is turgid, what happens? When it is flaccid, what happens? I really want you to employ that type of a knowledge to the content that you are placing here so that pretty much you have good grasp of it when you see it some other time. So I'm going to look for some more specifics, as it were, for the opening and the closing. I want to hear about the turgid and the flaccid as it is applicable. And I also I like the word stomata. I would also love to hear about those plants that close their stomata during the day and open it during the night. So this is for 3 marks. So we really don't want to just write one line. We want to make sure that we show that we have a good grasp of the content so that we can secure full marks. So let us see what's here. The guard cell controls the opening and closing of the stomata. Good way to start. When the stomata starts to lose water, all right, the guard cell close it as it becomes flaccid. This person, right, is spot on. So as it loses water, it will close the guard cell. So once it's going into water stress, it's going to close the cell. Now, the other thing is that we have some plants that they, sorry, just let me just clear up this part. I don't know how many persons know about CAM plant and C4 plant and C3 plant. That is probably a little more photosynthesis than this class. But there are most of the plants around us, they would have the stomata open in the day and close in the night. But when you look at some desert plants, they have the stomata close in the day and open in the night as one of the way to make sure that they maintain that water balance between the plant and the soil. All right. So I like the answer that is on screen. I shouldn't remove it yet. That is a nice answer there. I think that everybody wants to master the guard cell to that level where they can look at that to prevent water loss I really want to see the word flaccid coming out, because I want you to understand how the guard cell opens and how it closes in order to regulate the stomata. So you need to know that when it becomes turgid, it's open. When it is flaccid, it closes. I really want you to know that. I really want you to have that as a takeaway from that lesson. All right, so that brings us to the end of question number 1. It was a long day for question number 1, but I think we did well. In most cases, you answered the questions, and I did not have to say much. Now, we have a video for almost all the topics that we have done so far. We are now looking at cells, and we have videos on cells that look at as it is outlined in the CXC syllabus, because we only make videos for CXC biology, HSB, and grade 7 science. All right, great. So we're looking at figure 3, which shows 3 different types of cells, A, B, and C. All right, so quickly, we will need to identify these cells. So tell me in the comment section there, what is A, what is B, and what is C? What is A, what is B, and what's C? Okay. So animal, plant, and bacteria. I wish I understood that. Remember, we are dealing with different sets of learners. Some persons are faster than others, and we have to be mindful that what we're putting out, whether it is wrong or right, there are persons who are looking on, expecting me to accept or to make the correction. So you want to ensure that you put things out that everybody can learn from that which you're sharing. Whether it is wrong or right, we are learning. So A is going to be the animal cell, B is going to be the plant cell, and C is going to be... I could have told you prior, when I was looking at the question, I knew then that a student would have done this. You did not say animals. You did not say plants. However, you say bacteria. So which should it be? Should it be bacteria or should it be bacterium? I'm looking for that. All right. So I am going to look, and I'm seeing everybody saying bacteria, everybody is saying plant cell, everybody is saying animal cell, but it's like it's a Caribbean problem. Everybody is saying bacteria. So now I want to know, is it bacteria or is it bacterium? Because you are saying animal, you are saying plant. So why you would have said bacteria? Understand? And that is why we are not doing GC in the Caribbean, because you know you have the same problem that I have. All right? So it should have been bacterium because it is one. Right? So if you are going to say bacteria, then we need to say more than one. All right? But if it is one, we are going to say bacterium. All right? Great. All right. I like your answer. Your answer is not right or wrong, Pogs. I just want to point out the singular and plural. In my video, I did a lot on this to ensure that persons understand what is expected. Now, you need to remember these, A, B, C, and D, what they are, because they won't be named moving through this question. And for everything that we are going to answer here, you are going to need this information. Again, this is a repeat of the 2017 paper. And we're doing 2018 now. So look at the following question. Name 2 structures that are common to the type of cell. Let me see this. Name 2 structures that are common to the 3 types of cells. All right, tell me in the chat 2 structure that is common to all 3 cells. Da, 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 na, 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 na. Somebody's out of the block already? Wow, Jita. Oh, Jita, you're going to the Olympics this year. Where it is? I saw it just now. All right, great. So let me see what Jita is saying. Is it on the screen? All right. What happened? I'm sure I saw Jita with something just now. I'm gonna find it. Okay. All right, things common in all cells are cell membrane, cytoplasm, and mitochondria. Is that something that you saw? Things common in all cells, cell membrane. Interesting. Anything else? I was looking for this answer on the screen as being the popular answer. Cell membrane, cytoplasm. This answer, let us look at this answer. This answer is not correct because the bacteria does not have a nucleus. It's called a nucleoid. We call it a coil thread of protein, coil thread of DNA, but it is not a nucleus. We said that in the 2017 paper this morning, that the bacteria, it is a prokaryotic organism, which means that it does not have a nucleus that is membrane bound. Once it's a living organism, it's going to need some energy, so it needs to have some mitochondria somewhere, somehow, something to do that energy stuff. All right, good, great. Let's move on down. Cell membrane. No, nucleus is wrong. Remember, we said that nucleus is wrong. All right, so cell membrane and cytoplasm is always going to be the easiest thing to remember. Cell membrane, cytoplasm. Cell membrane, cytoplasm. All right, the nucleus is not correct. All right, if you observe, it's just a coil of DNA here, not a nucleus. And if you observe, you are seeing the cytoplasm there. If you observe, you are seeing the cell membrane there, and we are seeing the cell membrane there. We are seeing the cytoplasm there. We are seeing cytoplasm there, and this, of course, is the cell membrane. So those are things that are easily seen. You don't have to wonder whether it have it or it don't have it. You can just see it at a glance. You can see it. All right, let's move right along. All right, name two structures that, oh, we did that just now. State two difference between the type of cell labeled A and the type of cell labeled C. So want two difference between A and C. So write that in the chat for me. Remember what A and C is? What they are. So that's what it is. Two differences. So I think I'm seeing something here. So look at that answer on screen. A has a nucleus. However, C does not have a nucleus. Can we go again? I'm looking for the other one that just stuck out like a sore tongue. The flagellum. I wasn't thinking about that, but that's a nice one as well. Flagellum. We know that the bacteria, it has this flagellum that whips like the sperms and propel it, allowing it to move. All right, but I'm looking while the other cell doesn't have that. I'm looking for something else. We're not looking at the plant cell now, are we? Oh, somebody might be coming in late. I'm looking for something. Okay, great. I am looking for this, I think. Let me look at it on screen. cell C has a cell wall, A does not. Great. So great. So that's it. So if you look at A, it's the animal cell. It has a cell membrane, does not have a cell wall, but the bacterium has a cell wall. It also has flagellum that allows it to move. Remember that organisms move by swimming with a fin. Organisms move by walking. Organisms move by flying. But there are smaller organisms that move like by, for example, the amoeba, it moves by pseudopodia, false leg, and you have like some organism moving with cilia, and then you have flagellum. So these are just mechanisms that assist organisms in moving. We move on to the other question. Thank you very much for your input. It was really, really needed. All right, so we move to the other question, and I think we looked at this morning before we left as well on the 2017 paper. Now, tissues made up of type B cell, that's type B cell above, are placed in a container of concentrated salt solution and left for 1 hour. Describe 3 changes in the appearance of the cell after 1 hour in the concentrated salt solution and give an explanation to account for these changes. So we left the tissue tissue B, we left it in concentrated salt solution for 1 hour. We want you to tell us 3 things that we would have observed. So you are going to tell us the 3 things one time. 3 things that you would have observed. You would have done this type of a lab when you were doing osmosis with the potato strip. I'm sure that you would have done this, or if you were using the cucumber or something like that. So of course, you should have some amount of knowledge to answer this question. So let me just pop a look on this. It says cell B was placed in salt solution. The vacuole of the cell decrease in size. I want all 3 at the same time. All 3, because they are so close that you might say and overlap in some cases. It will become flaccid. That's a nice term because water from the cell was removed. So 1, it becomes flaccid. Water will be removed from the cell. From a high concentration in the cell to low concentration in the container. I realize that you guys don't want to use the term hypertonic. This is a hypertonic solution because the more concentrated solution is on the outside. Let me see what is going to happen here. I'm looking for something. Yes, it's osmosis. I am looking for something. Just give me 1 second. Sorry. You can turn on that for me, please? Can you turn on that for me, please? All right, great. So let me see some of the answers here. I'm going to look at Casanova. I'm just picking on it because it has a lot, so I want to see what is said. All right, the plant cell will become flaccid. So 1, it becomes flaccid. I'm still looking for something. Shrunken and small, so it will lose the size, it will become smaller. This is because water molecules from the plant cell is of high concentration to the container at low concentration, this is called a hypertonic solution. So great. I am looking to hear somebody at least say that the cell membrane will pull away from the cell wall. Yes, I know that it is flaccid, but I want us to understand that concept, that we understand what is really happening to a plant when the plant wilts. So I'm looking for the cell membrane being pulled away from the cell wall. Yes, the water loss is a notable thing. The cell becoming flaccid is a notable thing. And the cell losing size or mass is something notable as well. I'm seeing some other great answers coming up. I think we all did well just now. Thank you very much. We are going to be looking at the other questions. Let us just move on down. So all you need to do is to do your little explanation, make sure that we're looking at that. Now look at this question. It says tissue made up, this is the one we did a while ago. Okay, no. Tissue made up of type A cell and type B cell are placed in a container of distilled water and left for an hour. So this type of solution is a hypotonic solution because the concentration of salt is greater inside of the organism or inside of the A and B than it is in the water. So the higher water concentration is on the outside. Now describe how the appearance of type A cell is expected to differ from that of type B cell and give an explanation for this difference. Let me see what is going to happen over there. Remember, we did this on the 2017 paper this morning. We covered this on the 2017 paper this morning. Remember, I was telling you about the salmonella this morning? No, oh, wasn't about salmonella. It was about the salmon. Not salmonella, but salmon. Some person don't even know what is salmonella. All right, so let me look at what we have here. All right, I just clicking on the first one. Let me see if this is. Plant cell will gain water via osmosis and become turgid. The cell wall will help it to keep its shape. The animal cell, on the other hand, will absorb water, and since it has no cell wall, it will become turgid and it will burst, or it will rupture, or there's another fancy name for that cell being ruptured. If you know it, you can share it with us. All right, so that is pretty much what will happen. All right, so as Jita has it on the screen, that's pretty much what will happen. The animal cell will rupture, the plant cell will not rupture. We have completed that question, and I think the I is not on the paper based on what you told me this morning, and I yield. Oh, wow. Don't I like this question? I like this question because this morning I worked a similar question for you. So because I worked a similar question for you, I am going to ask you to work the entire question. This is a capital C here, and this is a common C. So I'm going to read the question for you, and you are going to do this genetic diagram. All that I'm going to do for you, because I want to make your life a little hard, I'm going to number that 1, I'm going to number that 2, I'm going to number this 3, I'm going to number this 4, I'm going to number that 5. I'm going to number this 6. I'm going to number this 7. And I'm going to number this 8. I'm also going to number here 9. So I'm going to make reference to those parts. So first of all, we need to read the question. Let's go through this question together. A colorblind woman mates with a man who has normal color vision. She's colorblind and she mates with a man that has normal color vision. Determine the probability of this couple having a child, having a colorblind daughter by completing the genetic diagram below. Now, if you observe, for the first time I am seeing it, CXC has actually set out the question exactly as you should have it when you are solving for the exam. So the first thing you want to do here, as usual, remember, is to define your alleles. Then the second thing, you are going to write the parental phenotype, and it is written on screen. Then the next thing you are going to write is the parental genotype. But now I want you to write the gametes in the comment section. Write the gamete for the male and write the gamete for the female. Write the gamete for the male. Oh, some persons out of the pack long ago. All right. Those persons who are a little behind, we are going to help you. No need to worry. No need to worry. We're going to slow it down a little bit. So 9. 9 is pretty much where the gamete should be. What should it be? So for 9, it is going to be, and let me just look at this one here. So for 9, it is going to be X with the C, but it is a capital C. I know that this is very small on the screen. For XY, it is a capital C. Remember, it said that it is a normal color. So this is supposed to be a capital C. X, capital C, and the C is superscript. I don't know how to write it here. And then we space it out, and then we write the Y. Then over on the female side, that's where we have the common C's. You have to make sure that you don't just write any old letter. You know exactly that they have meaning. So all that you're going to do is to space out the X and the C. All right, the C here being the allele and the X there being the X chromosome. Let us move on to look at what else is required of us. All right, I hope everybody would have understood. So I'm going over and I'm going to write it in because my student told me that that's what I should do. So I'm just going to do this. I am going to write for my gamete, I'm going to write an X here with a big C, capital C, and then I'm going to space it out here, and I'm going to write a Y. Now, by this, everybody know XY. Now, over here, I'm going to write a X, and this time, I'm writing a small c. This time, I'm writing a much smaller c, and then I'm going to write another X over here. You would have observed that it is spaced, and then I write another small c. All right, great. So you would have realized parental phenotype, remember 1. No, this is not even 1, because I want you to define the alleles when you go to the exam, because I don't want you to forget. So though it is here and blah, 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 you want to have it jot down somewhere you don't forget. All right, so that is 1, 2, 3. So now we have 4. And then we are going to move on down and we are going to populate this. So this is fertilization, or we could say segregation, or some person might say random mating, that is 5. All right, so great. Populate your table. Tell me what you are going to put in 1. Tell me what you are going to put in 2. Tell me what you are going to put in 3. And tell me what you are going to put in 4. So I want all of that in one message. what you're going to put in 1, 2, 3, and 4, put it in one message in the comment. What are you going to put in 1, 2, 3, and 4? I want you to put it in one comment. No, man, I am looking for, okay, so in 2, you are going to put Y, but you are going to put everything in one message. I don't want 2 or 3 messages for 1, 2, 3, and 4. I want to see, oh, yeah, okay. So 1, I'm going to put XC, and then 2, I'm going to put Y. Let me see that one. Let me put it on the screen. Who had that? Somebody started out. All right, let me look at this to see if this is one. So for 1, I'm going to put XC, and for 2, I'm going to put Y. So they are saying that they are going to put XC, and then they are going to put Y there. And then on this side, for 3, they are going to put X with a common C, and for 4, X with a common C. This is so correct. I think I really like that. Now, I want you to do the cross, and I want you, being that I have the answer, I think a lot of persons would have gotten this correct. Now that we have the answer, I want you to populate cell number 5. Let me see what this person is going to be. Cell number 5. Populate cell number 5. Write it in the chat for me. What is cell number 5 going to be? Or I'll just go on over and just fill it in as you said. Just let me fill this in for you. Some persons are not going to follow. So I think you said that I should put a capital, a big C here. And you said that I should put a Y there. And then you said that I should have, placed a X there with a little c. And I should place a X here with a little c. So great. So now you're going to tell me, what are we going to put in 5? Okay, so we're going to look at 5 coming up there. So, okay. So the naming convention has it that you are going to put the dominant allele first, so to speak. I think I clicked on something just now. So I think this would probably be it. I don't want you to solve for everything. We have to go slow. We are dealing with different learning needs. So if you observe here, we are using the big C up here and the small. So usually write the capital first, all right, the dominant. So I'm going in to do what I am told. So yes, that is correct. Oh, wow. So I am in now. So how did we get this? This big C came from up here. So we put that, and then we put our big C. And then we have this coming over where it intercepts. We go that, and then we're going to put in a little c. All right, great. So now I want you to solve for 7. Solve for 7. All right, so solve for cell 7. We are going pretty slow because we have, like I said, different learning needs. And what we're doing here is that those students who are more able, you are assisting us to assist those students. All right, so I am seeing another answer here. Let me try this one for 7. All right, it says, I don't know, where is that one? I'm trying to get this on the screen. I keep clicking on it and it keep moving away. All right, here we go now. All right, great. So for 7, we're going to be doing that again. So I'm going to go in and I'm going to erase the 7. And then I am going to write in What did we do? All that we did, we took this capital C with the X and we place it here with a big C, and then we take this here and we place it in with a small c, and that is solved. Could you solve 6 for me now, please? 6. I am going to go in and I'm going to change the color of the C just for those persons who are not getting it, that they can see where we are getting this C from. The big C, so I'm going to just change it out into another color. Not that it will affect anything, just that you can see clearly what is happening with the C's there. All right, so I'm going to use a blue. So I'm going to put a big C there. The C is the cap, the blue is the big C. All right, so the blue there represent the big C, and this represent the big C here. All right, great. So everybody know where the big C is coming from. Wonderful. All right, so let us see what is happening now. So we are looking for 6. So in 6, I'm seeing something right there. Beautiful. It is on screen. So let us go in and do that. So for 6, we are going to have, usually the naming convention says that we are writing the X before the Y. All right. So it is X with a common C and Y. All right. So all these that we are working on now is the genotype. Could you write A for me? 8 for me? Could you give me the answer for 8? What's the answer for 8? Answer for 8? All right. All right, great. There's 8 on the screen. I'm loving it. All right, great. So we're going to erase that, and we are going to write in 8. So what we do, we got this XC over there, X, C, little c, and this Y from here. And all that we have here is that which we refer to the genotype. Now, what I'm going to do, I'm going to prod you a little more. I am going to now use my green ink, and I want you to answer these questions for me. The question, I want you to tell me the phenotype of number 1. I want the phenotype for number 2. I want the phenotype for number 3. And I want the phenotype for number 4. So may I have a phenotype for number 1, please? May I have a phenotype for number 1? I want to ensure that you understand this concept of phenotype and genotype. May I have a phenotype for number 1? Number 1, what's the phenotype for number 1? Okay. No. Please answer only what is asked. I am searching for something specific. So I am seeing something that I like just now. All right, so look at the screen. Number 1 is a female colorblind. No, let us try something else. Let me see if I can find somebody with something different. Number 1. All right, look at this one. Number 1 is a female or girl normal carrier. If you observe there, it's heterozygous. When we are doing sex-linked inheritance, you must name the sex of the individual. So you have to name the sex of the individual, and you have to also identify the trait as well. So for number one, this person has the disease, will not be affected by the disease, but can pretty much pass the disease on to offspring. A male cannot be a carrier. Thank you very much. I don't think, Carlene. Thank you very much, Carlene. Could we solve for number 3? What's the answer for number 3? Solve for number 3 for me, please. All right, so it's a heterozygous female carrier. All right, so number 3. We're solving for number 3. Please solve for number 3. I'm sure I saw you just now. Well, we were wondering why you're Mitosis. We haven't seen you for ages. Yes, I'm going to load you up. Number 3. So number 3 is a female normal carrier. All right, female normal carrier, and she's heterozygous for the trait. All right, wonderful. We're now going to be solving for number 2. Tell us about number 2. Number 2, we're solving for number 2 now. You're watching CSEC Biology TCP. I am Mr. Wilson, and I'm going to be around with my friends until Tuesday night into Wednesday morning, where we're going to be doing HSB up until then. But I'm going to be with the bio students, I think, until Wednesday morning. All right, so we have work to do, a lot of work to do. I don't even know how I'm going to do all this because I have my personal work to do. I hope that they understand that I am doing the Lord's work now. All right, so we're solving for number 2. I have never really seen it being expressed as heterozygous colorblind. I've never seen the phenotype being expressed like that. Do I understand what it means? It is going to be, I'm going to run with this one. Number 2 is a male colorblind individual. All right, great. And if you observe, number 4 is going to be the same thing. All right, beautiful. So now that we understand that the letters represents the genotype, and interpreting the letters tell us the phenotype, now we are going to be answering the question that CXC actually wanted us to answer. So we are going to go back and we are going to read that question and see what we're supposed to do. It is saying that we are supposed to determine the probability of a couple having a colorblind child, a colorblind daughter. All right, so we are looking at the probability of this couple having a colorblind daughter. Do you realize here the importance of identifying the sex? Daughter. Look at that, because we are doing inheritance of sex or X-link. No, it's not inheritance of sex. It's sex-link inheritance or Y-link inheritance. All right, great. And I covered that this morning. So what is the probability of this woman having a daughter that is colorblind? So I'm going to come on over there and see what you have there. I hope that we understand. All right, I am waiting. I am seeing a lot of answers coming up. And I am proud of you guys. I am really, really proud of you. I'm really proud of you. The probability is 0. There is no daughter there that is colorblind. All right, I love that. So let me push you a little further. What is the probability of this couple having a son or sons that are colorblind? The probability of this couple having sons that are our son that is colorblind. Having a boy that is colorblind. What is the probability? Having a boy that is colorblind? What is the probability of a couple having a boy that is colorblind? All right, great. So once we have an incorrect answer, it's going to be a 2 to 4 chance, or 2 out of 4 chance. And if you have 2 out of 4 chance, you have to express it in its lowest form. So 2 into 2, 1, 2 into 4 go 2 times. So it's a 1 in 2 chance, or a 50%. So I'm going to show, because I realize that somebody got it incorrect. So when we get things incorrect, then I have to come in and clarify. So here, these are the boys that are colorblind, and we are looking at a set of 1, 2, 3, 4. So if 2 are colorblind, then it's going to be 2 out of 4. All right, and 2 out of 4 expressed to its simplest term is going to be 1 out of 2. Now, 1 out of 2 multiplied by 100 upon 1 is going to give us 50%. So it's going to be a half if we are using fraction. Oh, wow, sorry. It's going to be a half if we are using fraction. It's going to be 50% if we are using 50%. And if we are using ratio, it's going to be a 1 in 1 chance. Whichever way you are writing this, you must indicate what the 1 mean, or what the 50 means, or what the 1 in 2 mean. You can't just write 1 in 2, or 1 to 1, or 50%. You must say that there is a 1 to 1 ratio of whatever it is here. You are going to interpret the phenotype. Also be reminded that you could be asked to give the probability of genotype. It's not only phenotype. For those persons who are still struggling with the whole concept of phenotype, phenotype refers to the condition that you can see are the characteristic that is being checked. So if you are looking at me, my phenotype for skin color is that I am dark, I am a black person, and yeah, that can stay right there. All right, so what is the, okay, we did that. Probability of colorblind daughter, it is 0. So we would have completed that question nicely. You need to give yourselves a round of applause. I think you are doing really, really well. So if you observe earlier, I said to you, bio and HSB is somewhat similar. Look at that diagram on the screen. That is respiration. Now I'm going to show you another paper. Look at this paper, and I can show you several papers. This is the HSB 2018 paper. You remember when I told you sometimes the very year the same thing is on the paper? This is the HSB 2018 paper. And if you are looking at this paper, on screen is the 2018 bio paper. And in my hand is the 2018 HSB paper. And if you observe, they both have respiration, the same diagram on it. And that is why I continue to say, stop saying that Mr. Wilson is just looking at bio or looking at HSB. I pretty much have a fair knowledge of both syllabus, so I am sort of rationing it there. I do hope that I would have convinced you just now. All right, so let's move on to the other question, because respiration will not be on the exam. All right, so we're at question number 5. Now, this is pretty interesting. We started out the class this evening with components of the blood. All right, so you are supposed to name 3 components of the human blood that are important in protecting the body against pathogenic diseases. All right, and remember last night, I think it was, is it last night or this morning? We had a long session about pathogen. I would say that there are 5 groups. I would name them, and you were supposed to tell me the other one and you didn't, but I remember that you didn't. All right. Pathogenic disease, and state the function of each. So let me see those components of the blood in the chat that will be protecting us from pathogenic disease. So I'm looking on, you can start typing your answers. that component or those components of the blood that would protect us from pathogenic disease. When we started this event, I shared a video with components of the blood. I am going to share another video before the end of this class, again, because I don't want you to go to the exam and don't understand immunity. All right. All right, so I'm seeing an answer here, so I'll just grab it. It's on screen, on screen. So lymphocyte, platelets, phagocyte. Lymphocyte, platelets, phagocyte. Please be reminded that the white blood cell is called leukocyte. The red blood cell is called erythrocyte. And the platelets are called thrombocytes. All right? Great. So there we have the answer on screen. That was easy for a lot of persons. I wish I was bright like you guys. Now, what we're supposed to do now, we're supposed to state the function of each. Sometimes we're forgetting to do that. We're not remembering to do that, but we need to state the function of each. All right, so let me see the function now. Da da dee, da da do. So somebody's saying here, I always animate and talk some little stuff when I go to class. Yeah, I like to tell stories. I'm a storyteller. So I'm teaching and I'm just telling you a story. But if you listen to my stories, it's actually the lesson that I'm teaching. All right, so there we have and I'm going to tell you my story before I leave. Lymphocytes, they produce antibodies. Great. All right, so I continue to look. I'm seeing lymphocytes. No, somebody's saying something, not telling the truth about phagocyte. Phagocyte would not be happy to hear you say that about him. You can't tell such a lion on the phagocyte, on the lymphocyte. All right, Cody, careful. The lymphocyte is not too happy with what you said about it just now. All right. So the phagocyte, they are going to engulf and digest pathogen. All right, so we have placed the phagocyte on screen. We have placed the lymphocyte on the screen. What about the platelets? What about the platelets? What's the function of the platelets? So Amaya, I was telling them that you taught me about the tongue dispenser when we were looking at signs and symptoms this morning. Okay, let me see something here. Platelets, something is on the screen from Casanova. That's not it. All right, platelets used in the clotting of blood. Okay, all right, great. So this is my story when I am really in my animated spirit. So you fell off your bike, you got a cut. When you got this cut, the first responder, which is the lymphocyte, came on the scene. Now, while the lymphocyte was on the scene, the plate was pretty much securing the area. The platelet is like those police that come. They are not CIB. They can't do the investigation. All they do is to just secure the area. So the platelet was pretty much securing the area during that time when you fell from the bike. And while you fell from the bike, then you had the little investigators came on the scene, which are the lymphocyte, and they are which are the phagocyte. The phagocyte come on the scene, and what the phagocyte will do, they'll try to eat up any foreign matter that invaded your space. But if the phagocyte can't manage all that is happening, it's going to just take its phone out and it is going to call for lymphocyte. Now the lymphocyte will just fly in on their helicopter and then they'll just start dropping these missiles. Antibodies, they're going to just drop that. It's like a warplane, you know. I'm sorry, I'm beside the road. If it has caused so much damage to me, I wonder what about him or her? So what happened now? It's a car playing on the outside. What happens now? So that is my little animated thing. So it is like soldier going to war. You have the ground troop, which would have been the phagocyte, doing the work. But if it's too hard for them, then they have support system of these airstrike, which is of course going to be the antibody from the lymphocyte. The lymphocyte in most cases is going to go there, but you're going to have more coming there if the phagocyte really can't treat with it. While the platelets secure and try to seal the wound so that there is no further invasion of foreign organism. Very good. Let's move right along. All right, great. Are you ready for my video, or are you comfortable answering this question about immunity? Are you comfortable answering immunity, or you want to share the video with you? Oh, yes, thank you very much, Tori. That's why I write. That's why you need to see my book or read one of my books. I write. Everything means something for me. All right. Okay, great. So I am going to share the video with you. I'm not going to share the video again after today. There are other things that I'm going to need to share, but you want to try first. All right, great. Go right ahead and try. I hope you can read. If you can't read, I'll read it for you. All right, just give me a second. Let me just get this off the screen. All right. Share the video, please. All right. I shared this video this morning. I think I started my show this morning with this video, but I'll share the video with you. And this. And remember that you can go to my channel to watch the video. Though I'm playing the videos from my laptop, they are actually on the channel. So we're nearing the finish. We're at question number 5, and we're going to be going next to question number 6. And at question number 6, that's going to be for the night. And I do hope that I'll have some time left in the night to do some more work. But I delayed the show today because I observed that more persons are available this time of the night. So it was deliberate not starting the show when it was scheduled to start at 7 instead.
Production. Today we're looking at types of immunity, and of course I am Mr. Wilson from CSEC Biology TCP. There are 2 types of immunity, active and passive. Let's look at the animation for active immunity. Active immunity occurs when an individual is exposed to a disease which causes the immune system to produce antibodies specific to that disease. It is important to note here that the individual would have been exposed to the disease,
no problem,
and that immunity is of course being developed as a result of action within that person or organism. So here we have a fellow being introduced to the ranch, and with him he brought pathogen, and you're seeing the pathogen populating the screen in red. And as the pathogen enters the environment, you're seeing that the lady is developing some antibodies that are of course going to be guarding her against this particular pathogen. So of course she will not become ill as a result of her building her defense from within. So here she's exposed to the disease, and as such she would have developed some resistance. With passive immunity, what happens is that the antibodies are of course given to the individual rather than them producing it through their own immune system. So in the passive, you're seeing that it is given to the person, while in the active, it is the person who is actually making the antibody. So in passive, the antibody is made elsewhere and given to the individual. However, in active, it is of course made within the individual. And we're going to continue to reinforce that throughout this video. Here we have examples of passive vaccine there we're seeing, and of course a mother with the fetus, and we're seeing a mother breastfeeding. Now, immunity could also be considered as vaccine there we're seeing, and of course a mother with the fetus, and we're seeing a mother breastfeeding. Now, immunity could also be considered as natural or artificial. With natural immunity, it is organic. Natural innate active immunity. This is where we start now to think about our exams. Now, these words are all words that they carry specific meaning and of course direct you to the particular immunity. So natural, it is organic. It has no influence of man. Now, this type of immunity is an immunity that you are born with. How do we know you were born with this immunity? It is innate. That suggests that you were born with the type of immunity. Now, where do we get the antibodies from? Active here tells us that you are making the antibodies. So whenever we see active, it means that the antibodies to safeguard you against that particular pathogen is made within the individual and not with external assistance. Then there's also natural innate passive immunity. And let's decode. Natural, it's organic, it has no human interference. Innate, meaning that it is made within a organism. And of course, passive here mean that it is given. It's something that is given. It is passed from one person to another. When passive, we talk about passive, I'm sorry guys, I just paused the video because I realized that there was some chat in the room that was a little disturbing. So I just paused the video to block the individual because it's pretty much a learning atmosphere, and that distraction was more than I think that we could have handled. Active, it means that the antibodies to safeguard you against that particular pathogen is made within the individual and not with external assistance. Then there's also natural innate passive immunity. And let's decode. Natural, it's organic, it has no human interference. Innate, meaning that it is made within a organism. And of course, passive here mean that it is given. It's something that is given. It is passed from one person to another. When passive, we talk about passive, you are given the antibodies rather than you making the antibody. So here, the offspring gets the immunity from the antibodies that are, of course, produced by mom that crosses the placenta to the fetus. So the antibodies are not made in the baby. The antibodies are actually made in the mother and of course passes the placenta to the baby. The baby also acquires some amount of immunity from the breast milk. So passive here suggests that you were given the antibodies. It's not that it was made within you. So the baby was given the antibody. It's not a case that the baby is able to make the antibodies for itself. Now, innate, of course, when the baby is born, or it is made within. Innate, when the baby is born, will born that with a short-lived immunity, and it is, of course, innate speaks to being within the individual. Why is this youngster running? It seems so happy. Of course, he would have acquired acquired active immunity. What does that mean? Acquired, the immunity that develops after birth. So immunity, if we were supposed to compare, we'd compare acquired to innate. So if it's from birth, then it's innate. If it's acquired, then it's after birth. If we were supposed to compare natural, it would be compared with artificial. If we were supposed to compare active, it would have been compared with passive. So this type of immunity is one that develops as a result of exposure to the disease. Example, smallpox and tuberculosis are examples. Now, this youngster probably would have been exposed to the disease, and having exposed to the disease over time, he would have gotten some immunity. So there's a great possibility here that he is now told that he's no longer suffering from the disease, he's immune to the disease, because his body would now be making the antibodies to fight off the particular disease. Who wouldn't be happy? That's what most persons would be saying. Now, artificial immunity is one that is, of course, introduced, or it has direct human influence. So whenever we go for vaccine, that is a good example of artificial. Now, it could be artificial acquired active immunity. And let's break this down. Artificial meaning that it has some human influence. Acquired meaning that it was, of course, developed after birth. So you were not born with it if it is, of course, acquired. And then active, even though it was given to you by a human, it is within your body that the antibodies are going to be made. So with this type of immunity, artificial acquired active immunity, immunity that developed as a result of individual being given a weakened form of the disease. And the disease here is weakened by eating, or we could use formalin, or we could re-inject several times, and of course weaken the pathogen to a manageable form that can be injected in someone. Usually it is administered through vaccine. Examples would have been that for tuberculosis, polio, and BCG that we would have received in the early years of our life. It's very important for you to pay attention and look at the photographs that we're using in the pictures, as they are giving you prime examples of each of the type of immunity. Now, artificial passive acquired immunity. Artificial here, human influence. Passive meaning that the person was exposed to the antibodies and not to the disease. So here the person would have been given the antibodies because it's passive, and acquired meaning that this resistance, this immunity actually developed after birth. So we have both on the screen, and we continue to emphasize that when you get to the exam, you can break down this long name, artificial passive acquired immunity. What does it mean? And if we were supposed to see passive acquired immunity, what does it mean? And if we were supposed to see passive immunity, what should we understand from this? So here we have broken it all down. We have given it a meaning for all the words in the names. So you should be able to map this nicely when you get to the exam. Now you're seeing this, and this one is synonymous with bites from snake. Now it is acquired through the introduction of antibodies directly into the individual affected by the disease. And usually you would have seen my first video on serum and plasma. Here we have what is called antiserum, which is used to treat this type of problem. It is fast acting but lasts for a short time, and it is used during cases of emergency where somebody is bitten by a snake or where the person is suffering from tetanus. This type of a vaccine is administered so that there is quick response and the person could of course recover quickly. If you look at this young man, he was bitten by a snake probably, and of course he's been injected probably with of course antiserum to ensure that he will survive. So looking at the snake, it reminds us that it is artificial passive acquired immunity. We want to ensure that we are paying attention and be reminded that you could go over the slide however many times you so desire, so as to make sure that you understand well that which we have covered. We have just looked at what's the difference, and we're looking at type of immunity, or one might say types of immunity. I am Mr. Wilson from CSEC Biology TCP for TCP Academy. Please be reminded to like, share, and of course, subscribe. And when you do, click on that notification bell and select all so you'll be notified as soon as there is a new video.
All right, great. So we are back to the question now. I will share in the question. Let me just check to make sure that you're hearing. Can you hear me? Can you hear me? Can you hear me? Oh, great, great, great. All right, so we are back to the question. We are going to be bringing a wrap to this now. So let's read this question. Marianne, a 1-month-old baby, acquires immunity from her mother through breastfeeding. Marianne's father accidentally punctured his finger with a nail but does not need a tetanus antiserum injection because he received a tetanus vaccine the previous year. Explain how the type of immunity that Marianne acquired for breastfeeding differ from the type of immunity that her father acquired from his vaccine. In your explanation, state the name of each type of immunity. So the first thing we want to look at here is the type of immunity, and then you can give your explanation. So let me see what you have there. So what type of immunity Marianne the baby got? What type of immunity? So the father had artificial, active immunity. Let me just up and back. Let me look at this one. The type of immunity shown in breastfeeding is natural passive immunity, while immunity to the father was acquired from vaccine, which is artificial active immunity. So it's pretty much an artificial, that's correct, artificial active immunity. Now, how this works is that babies are not producing antibodies, so the antibody would have been produced in the mom and it is passed on to baby. While for the father, he would have been given a weakened strain of the pathogen, and it is weakened, like I said before, by formalin or it could have been weakened by re-injecting, or it could have been weakened by heat treatment, and of course, that would have provided the antigen for the body to make the antibodies to safeguard the father from tetanus the next time it comes across this particular disease. All right, so we move on to the other question. I'm realizing that persons are really anxious. I am seeing PMAT being mentioned in the chat, but it's if you're going to do well in your exam, one of the things is that you need to be disciplined, right? If we're not disciplined, then we are going to be having problem. We're going to do things just haphazardly, and then we probably don't get as much as we should have gotten from the opportunities that were available. All right. So the authorities, look at this one. The authorities prevent children who are not immunized from registering in public schools. Suggest two reasons why some parents may have a valid concern about immunizing their children, and two reasons why the authorities maintain their position and preventing the entry of unvaccinated children. So this is pretty much a social issue. And if you are doing HSB, I think this is more of a HSB question, but it is on the biology paper, so we have to treat it. So let me see what you have for this. Two for the parents and two for the government. Why the parents should maintain the position, why the government should maintain the position. Let me hear those persons who are on the side of the government and those persons who are supporting the parents. Those persons who are supporting the parents versus those persons. You want me to read it over? All right, so I'll just do that for you very quickly. The authorities prevent children who are not immunized from registering in public schools. Suggest two reasons why some parents may have a valid concern about immunizing their children, and two reasons why the authorities maintain their position on preventing the entry of unvaccinated children. So you are going to be given those two reasons that the parents have that you are agreeing with, or that is plausible. So let me look at what Casanova is saying here. All right, so reason for parents, they could have religious belief and they are uncertain about the vaccine. I think Casanova is spot on. Religious belief, we have heard about the cases of the Tuskegee experiment. I don't know if you are too young to know about that, or if your teacher would have passed that in ethics in science. The Tuskegee experiment, where persons were injected with gonorrhea and they were given placebo as treatment for the gonorrhea. They did not know it, and that was an unethical experiment. So persons are not trusting the system. They think they might just give them anything and tell them anything. So they are given X and then they are told Y, when pretty much it's much more than that. So a religious belief for the parents, and of course, they're uncertain, or the culture doesn't allow them to swing along with the government. Let me look at another one. So that is spot on from Casanova for the parents. Let me see if there's anything else for parents that might be different. One reason why some parents may have a valid concern about immunizing their children due to the fact that deliberate introduction of a weakened. Oh, but a lot is said here. We have to try to sort of say it less than this, or else we are not going to finish the exam. So I want to tighten up on those words. All right? I just try to make it spot on so that we are not crying that, oh, if I only had 5 minutes more, it would have been over. But I understand where you are going. All right. So I am seeing a lot for the parents. Let me see what Amoy has. Immunized children, who are you answering? Who are you supporting, Amoy? That is very important for you to share that with us. We want to know who you are supporting. All right, so 2 reasons for the government. Concern about the ease of contracting this from contracting or spreading the disease, and they will experience all symptoms. They are concerned about mutation. I didn't think about that, but that's plausible. Concern about mutation of the disease in the unvaccinated person. I like mutation. I really didn't think about that. That's a nice one, I think. Really, really nice. I didn't think about that one. All right, so I am seeing some nice one here. Let me look at this one again. It says here, parents have different religious belief. They could have cultural differences, and they are uncertain about the vaccine. Yes, this is wonderful. All right, great. So I am seeing some answers that are really plausible. So one, the parents might have religious or cultural belief or knowledge from history with vaccines and what was done. The government has a right to protect its space and its citizen. Anything that has no owner or that is unregulated suffers from what we refer to as tragedy of the common. Now, when anything suffers from tragedy, anything that suffers from tragedy of the common is referred to as a common good. Now, it is the state's responsibility to protect common goods, rivers, seas, park, the atmosphere. Now, if we have a disease going around, it is the state's responsibility to ensure that persons are protected against themselves and protected against others. Now, so you really don't want this disease to be passed on. The government wouldn't want that for other persons to become infected because it's going to put a strain on the healthcare system. Now, putting a strain on the healthcare system will also put a strain on the family. Putting a strain on the family will also reduce work time for company, and companies are going to be missing deadline, and then there's going to be an economic backlash. Now, when we're having an economic backlash, there's a great possibility that we're going to be having a social unrest in the country. So the government has to think way ahead as to how the problems that could arise as a result of this type of a thing. The other thing is that the government must maintain a budget, and they really can't spend out money just to buy medication because persons were not as responsible as they were supposed to have been. So I think you did well there. Yes, good. Mutation. Yeah, mutation is another thing. The disease could mutate if it's not controlled. So we want to ensure that that is also addressed. I think we are doing well so far. Let us just move on now into question number 6, which is the final question on the paper. I'm not too sure if you did this, but I saw somebody started earlier in the chat. Now is the time for that person to really step up front and assist us with answering these questions, because I realized that the person was really anxious and was pretty much distracting others. So now it's your turn to share with us. We are now paying attention to you. So you can take the stage and let's address mitosis. All right, great. So this particular question, it wants us to look at we are supposed to describe how the process of mitosis results in the production of identical cells in asexual reproduction. Now, like I just said, the person who was treating with this earlier, I invite you to rejoin so that we can give you your chance to share. We really want to hear from you. The only concern we had then is that things must be done a particular way. Order, discipline is very, very important. All right. Regardless that the space is free, regardless that you can choose where you go, wherever you go in your life or in a common space, discipline is very, very important. All right, great. So just let me just continue until the person shows. All right, so we use this to represent mitosis. So you might have seen somewhere PMAT, M, A, T, and you would have seen a C there. So it is IPMAT-C. Now, mitosis is actually just this part that I'm going to circle, the PMAT. This is the only part that represents mitosis. This part here that says PMAT. So that's mitosis. But before mitosis, we have interphase, and after mitosis, we have cytokinesis. Mitosis is actually prophase, metaphase, anaphase, and telophase. In former years, CXC would have just asked you to name phase 1, phase 2, phase 3 are stages. But I really think that it's more important for you to understand the names of the phases and what happens in each phase. Now, when I teach this, just before we answer the question, when I teach mitosis, the first thing that I tell my students to do is to identify mitosis goes through the process also known as PMAT. All right, great. The first thing I say to my student is try to identify the parts that you really don't have to study. Now, the parts that you don't have to study is pretty much big. It starts with metaphase. That part you really don't need to study. Because with metaphase, you are going to find the chromosome lining up down the center of the cell, one would say on the spindle fiber. So once you see the line up along the center there, you know that that is metaphase, right? Now, if you look at the word a, look at the word anaphase and the word away, once you are seeing the A, it means then that the chromatids are moving to opposite end of the cell. They are moving away. So now that we have taken care of metaphase, where the chromatids line up along the center, and we have taken care of anaphase, the chromatids are now moving away to opposite end of the cell. We now know that after anaphase, what is taking place is telophase, and what is going to happen is that we're going to be having some reforming of the nucleus, reforming of the cell. Now, we know that that is what happens at the end, and at the start where we have prophase, we know that the chromosomes would have become shortened and thick and all of that, so we know that bit. But how do we answer this question? It's a very long answer. It's a very long answer, really long. I don't know if you could answer that in the space that we have there. So pretty much a cell division is, with this type of a cell division for this particular question, the cell division is passing on the diploid number of chromosomes. Remember, cytokinesis is after mitosis. It is passing on the diploid number of chromosome, and we know that we use N and 2N, 2N representing the diploid and N representing the haploid. So the process starts with pretty much telophase, with prophase, where it makes two chromatid are formed. And I'm giving you just a condensed form of what it is. Two chromatid from the chromosome. Now the process continues from prophase into metaphase, where I said earlier, the chromatids, the chromosomes, they are lined up along the center of the cell on the spindle fiber, or one might say the middle of the cell. Now, after that, they are going to get into the anaphase, where they are pulling away. And then we're going to get right after that into telophase, and I'm giving you a real condensed form of the answer here because it's not a really long to write. All right. This is 2018 paper. All right, so In telophase, we are going to have the two nucleus being reformed, and the membranes around the chromosome will start reforming. After which we are going to we're going to have a membrane coming down through the middle of the two cells. Because with mitosis, what happens is that we have two daughter cells being formed from the process. So that is, don't do that. That is how we end up getting That's how we end up getting identical offspring from the process of mitosis. Like I said, it is pretty much long for us to answer here. So I suggest that you revisit this particular question in your textbook and on the paper. However, there is another question that I think that you could do very easily. And it's a question that follows. So you see, this is like an entire page that you are supposed to write. It's sort of too much for me to ask you to write in that little space there. But look at this question. Plants that reproduce sexually have some advantage compared to those that reproduce only asexually. Outline 2 advantages and 3 disadvantages of sexual reproduction. So we are looking at sexual reproduction, and we want to look at some advantages of sexual reproduction and advantages of and disadvantages of asexual reproduction. So just type that in the chat there for me, and that takes us a little closer to the end of the paper. We have just 2 more questions to answer after you would have successfully answered this question. All right, so I'm sorry, I got a little distracted by what's been said there. All right, advantage, we have variation, because you would understand that we have random selection in mating. Then we have random fertilization, as just about any sperms can do the fertilization. And then we have crossing over, and all of that bundled together would, of course, give us the random fertilization. Advantages. All right, so we have variation, a whole lot of things happening all over the place. So let me just get into it. So we have variation in the species, that's an advantage. We have the species providing that it's from a desirable stock, they are better able to adapt to changes, and this is as a result of variation. Now, metapopulation can be formed easily. What do we mean from metapopulation? If you were supposed to have a mango and you go down the road and you toss the seed, then pretty much we are going to be having a new mango tree. If you toss an apple tree, and we're going to have a new area where we have this plant being grown, we refer to that as a metapopulation. Now, the offspring may be better, maybe of better quality, because with sexual reproduction, once you know that women tend to want brown babies, because if you are brown, you stick around, and if you are black like me, you go to the back. So they are going to look for the browner fellows to be their children or child's father. And if they have curly hair, they want the guys with the curly hair. So you understand that in this case, they are looking for offspring that may have a better quality, as they would have it in the human race or in our local persons. Women tend to do that. While the men, so I'm not only going to hit the women, the men now want to find women who have a very wide hip and they are really curvaceous. And one of the things that men want to find, I'm going to go back to women. One of the things why men want to find a woman with that type of a shape is this thing that is hard coded in our phylogenetic memory that says that if a woman has a wide hip, she's going to be able to have easy passage or pretty much deliver a child very easily. Now, a woman is looking at a man and she'll tell you about he should have six pack and he should have a chest that is well curved and the pectoral girdle should be wide. Now, this is hard coded in the woman phylogenetic memory that if the man possesses this type of attribution, then it means therefore that this man is going to be strong and he'll be able to protect her and take care of the child. So there are some things that are just hard coded in us. We don't know why we do it, but we continue to do it. So a man will continue to look at a woman's buttocks once she passes him, and that is pretty much what is happening, and he's not even conscious. A woman will continue to look at the man's chest to see is he strong enough? Is this man worth it? That type of a thing. Now, a disadvantage of sexual reproduction is that a lot of time must be spent searching for mates. You understand that if you're as dark as I am, you probably wouldn't have as much luck as a fellow who's brown. So I probably have to talk to that girl and, hi, I don't like you, you're too black. And I talk to another girl, hi, I don't like you, you're too brown. And that type of thing. And then you have to invest that whole lot of time trying to find a mate. Then there is also the slow reproduction of offspring, because I want to have a kid within the next 9 months, but I just can't find this mother to mother the child. So you understand that I might go for the rest of my life searching for Miss Ideal and she just never come along. Then there is also few offspring that is produced. So the production of offspring is slow and only few offsprings are produced from sexual reproduction. So that makes it a disadvantage. Now, the offspring may, of course, be of undesirable trait, because the truth is, while we teach you here in biology that when you are going to find your partner, you should make sure that you do some genetic counseling. In most cases, our heart does the thinking rather than our brain. So you go out and you find persons who have undesirable traits, undesirable genes. In some cases, they are not even sharing with you what these undesirable traits are. And before you know it, you would have changed your wonderful gene pool and adding something to your family that you really don't want. I am not saying that persons who have their various ills should be single, but I am also asking myself, why should I take up more load than I can carry? All right. We are a very good friend. I really like you. I will do just about anything for you, but we just can't cross this line. And you understand there are many females out there and many males out there. We never cross line, but we are good friends, and so too you can be my good friend. You have this problem. I don't even need to tell you that. You know whatever problem you have, and it's no discrimination. It would be more discrimination if you ask me to hurt myself, right? That would be a greater discrimination. So we move on to the other question. I would have said a lot there, a little bit CXC and a little bit beyond CXC, but like I continue to say, I think you should know a little more than CXC. If time would have allowed me, I would have taught mitosis and meiosis, but there are many videos out there that nicely covers this content, so there's not enough time for me to do it, so it makes no sense. Now, scientists have successfully cloned animal species. We would have heard about Dolly the sheep. So there's two reasons why human may choose cloning, may choose to clone animals. Now, what this question is really asking, what is this asking? The underlying question here is to identify importance of asexual reproduction. If you can decode this question, it is asking you pretty much to identify importance or reasons for asexual reproduction. All right, take short period and okay. So let me look at this one, Jody and Campbell. We have one question to go. It takes a short period of time and two can produce a lot. All right, very good. This is good. Can produce a lot in a short time. So this is very, very good. Persons might choose cloning because they will, of course, another thing is that they will produce superior species. So that is one of the, this is not cloning though, but when you look at the production of the mule with the donkey and the horse, it's a hybrid that they want, with a superior quality to carry load, a higher animal that can carry more load and move a little faster than the donkey. So with cloning, an advantage of cloning is that it is economically friendly because you can really get what you exactly wanted. And of course, you, as I said, you can have superior quality in your stock. So that makes it very, very important. That takes us to the other question, which is asking us to, and this is going to be the final question for the evening until you see the videos. Outline 2 ethical issues related to the cloning of human. 2 ethical issues that are related to the cloning of human. Just 2. Oh, you are 20 minutes behind. I'm sorry. All right, good. So when we look at some of the ethical issue of cloning human, in many countries, it is illegal. It is illegal in many countries. Some of the risks that are associated, I'm seeing what's happening in the chat. Some of the risks that are associated, yes, religious is a big thing. It removes individuality. That's another nice one. Persons are playing God. That's another nice one. It could lead to some form of mutation, cause persons to age faster than they should age. Continue. It could decrease the value that we put on human. Could decrease the value. So can you imagine, because I really like Mr. Wilson, I created another of Mr. Wilson. That wouldn't be nice. It might be something that is exploited. We have to look at that. We have to look at that very seriously. And of course, it could cause, like I said earlier, faster aging. But let me look at this particular question. This particular answer. I wanted to look at this one. Some people don't believe it is right to have someone walk around with your genetic information and that the person can commit crimes and make it look like it is you. So it is not something I think that is well explored. One, and we are not too certain what will happen. We have seen all these sci-fi movie where persons try to create this and create that, and then we end up with a mutation, and then we can't control the mutation and all of that. So the risk factor in this is way too high for us even to consider it as being a viable option. Now the risk option with cloning, it's a possibility that when you are cloning an individual, the person might lose their life because the high risk that is associated with it. So we have to take all these in consideration before we think about cloning. My greatest fear is the mutation. Can you imagine we have now a new animal in Jamaica or in the Caribbean that will just walk around and eat people? It is resistant to gunshot. It is resistant to bomb. It is resistant to poison. Then you know what would have happened? Then the entire human race, after some time, would have become extinct. So we really don't want to play God and make anything that we can't control. Great. This takes us to the end of the show for this evening. That was the end of the 2018 paper. Tomorrow, we are going to be doing the 2021 paper for the bio student. And on the 2021 paper, we have the heart for those persons who have seen it. One of the diagram on it is the heart. And we have the feeding relationships that is also on it. And I'm seeing something here. I think I might have it on screen. No, I don't. So we have a couple of things to do here. That graph from photosynthesis with the limiting factor It is on the paper as well. And the internal structure of a leaf is on the paper for you to label as well. So it has photosynthesis coming on. Now, I don't know if you realize for the paper we just did, it is the only paper that we have done so far that has plant reproduction on it. So we have looked at 6 years worth of past paper for biology, paper 2, and the 2018 is the only paper that has plant reproduction on it. All the others look at something that has to do with the female reproductive system and contraceptive. Now, I was looking at the chat earlier and I realized that some person said that what was promised for geography was not on the exam. Now, I really don't know, but what I found a little startling was that the date on the exam paper did not change. So the exam was actually slated to be May, June, and that is the paper that came out. Now, I don't remember well when the broad topics came out. But I'd hate to know that CXC sent out a document to say that you are not doing this, and then at the end of the day, it appears on the exam paper because they gave us their words and we are preparing because we want to do well. We really don't want to put our life on hold. All right. So that is going to be it from me. I'm not too sure. I am not going to be on in the morning. Definitely, I'm not going to be on, but I'm going to be having 2 shows tomorrow. So you want to like, share, and subscribe so that as soon as I send that thumbnail out, you'll be getting it. All right. You want to ensure that you do that. You want to invite other persons to join. You want to give it some love. And please be reminded if you need to send us some donation or your church or wherever want to donate to us, you could do so by sending us an email and we can make the arrangement as to what will happen. Let me see, I'm seeing some comments coming in here. We're at the end of the evening. Or what time tomorrow? I hate to give time zone because based on my workload, I think for the bio students, based on what I have seen, you do better with a 7 o'clock class. I have realized that. I have realized that at 7 o'clock, you are all here. I know that you don't do well with an early morning class, but we are going to be having 2 classes tomorrow. So there's a great possibility that your class will start at about 7 tomorrow, and that's 7 in the evening. And the HSB paper that we'll be doing, which I don't know which one it is as yet, but we'll be doing an HSB paper. The HSB paper will start a little earlier. Now, after we would have completed the paper tomorrow, we might go into looking at some of the broad topics and what exactly we're supposed to be looking on and some of the problems that you're having. If we can have some of those addressed, we'll try to treat with that. The exam is Monday. If I happen to get a copy of the paper on Monday, I will make the answers available Monday evening. In a matter of fact, I was summoned by my student that the answer for the paper must be available Monday evening. All right, thank you very much for joining. I hope that you will all do well. I trust that I assisted you as much as you needed to, and you are now good, ready to go. My HSB students who have always been here and bio students who have always been here, I continue to love you just the same, but I know you understand that I have to share the time in catering for these students to ensure that they can go and ace the exam. You are going to be getting your live session starting tomorrow, Monday, and Tuesday. All right? Great. Enjoy the rest of your evening. Good evening. I really don't want to up on into what teachers did or did not do. What we can tell you is that we have a paid channel, and on the paid channel, you will never get an opportunity to say that we did not, because we have several videos there. We have the entire syllabus in video, and we have several animations. You would have seen many of our animations. So For those persons who are not so much text, in some cases, you can find the video for a particular topic on the channel. So we have done a whole lot of work, whole lot of work. We have listened to the advice of the students that we have been working with. And for that reason, we are sure that we are in good contention to offer a great product. We strongly believe that we are in good contention to offer a great product. So persons who need to do this can sign up on TCP Academy to get some assistance. That's going to be it for me this evening.
Looking at the properties of enzymes. In our previous lesson, we looked at what are enzymes. Just a reminder, enzymes are biological catalysts which actually speeds up the rate of a reaction. Important to note is that enzymes are made in the cell, and of course they are all protein. And here we have the elements that protein is made of: carbon, hydrogen, oxygen, nitrogen, sulfur, and of course phosphorus. And the cell there, just a place where the enzymes are made. The first property of an enzyme is that enzymes are specific. Here we have carbohydrates acting on starch, and carbohydrates will never act on protein. Likewise, protease will never act on starch. Now, one of the reasons for that is that enzymes have what we call active site, and here you're seeing an enzyme, and of course the active site. Now, the enzyme here will only, of course, act on this type of substrate, because this type of substrate will nicely connect to the active site of the enzyme. So we see here that enzymes act on only one type of substrate. They are specific
The
in
HSB
their
exam, I'm thinking
action,
that it
and
is
that
on
is of course
Wednesday.
one
Ask the persons in the
property
group,
of enzyme.
what is the
Let's look
exam?
at
I really
another.
don't
The
know.
second thing is that enzyme, their chief function is of course for chemical digestion, and of course they break down large bonds into smaller units. Look at the example here. We're breaking down starch, and of course amylase is breaking those big bonds of starch into smaller bonds called maltose. And of course maltose is acted on by another enzyme called maltase to eventually break it down into glucose, which of course the body will be using as a monosaccharide. So here we say for 2, this is the second property, enzyme breaks down large bonds into, of course, smaller unit. Now, the third thing we want to know, of course, a third property of enzyme, is that enzymes are not chemically changed at the end of a reaction. And you'll remember our enzyme from our first slide looks pretty much
All
like
the
this, where
videos
the
can
active
be
side,
found on
and
my
it is
channel.
of
So if
course
you need to
acting
watch a
on the
video, you could just
substrate
go over
as
to the
seen
channel
on
and choose the
screen.
one you
So
want.
here we look at the enzyme combined with the substrate. Of course, the bond is broken there, and you will realize over here the enzyme will of course release the product, and there the product is. But look at the enzyme remain pretty much the same. The enzyme is of course chemically unchanged at the end of the reaction. So there we have the third property of enzymes, and we move on to the fourth property. Enzymes work best at a particular pH, and we refer to this pH as the optimum pH. While the enzymes might be working in pretty much a range, as for salivary amylase will work at a pH of about 7 to 8, and pepsin pH of 1 to 3, and trypsin, which is of course found in the small intestine, will work at about 7 to 8. But if you look at the graph over here, you're nicely seeing it intercepted in the middle there, and it shows pretty much the optimum pH, and that's pretty much the best pH, or where the enzymes will perform at their best. So the fourth thing here, just a reminder, enzymes work best at a particular pH. You want to ensure that you're following the lesson because you're going to be having a little quiz at the end of the lesson. And we'll move on to 5. Enzymes work best at an optimum temperature. And that's about 37 degrees Celsius. That is pretty much the human body temperature, somewhere around there. And of course, you're seeing the graph again showing, of course, temperature and of course the rate of reaction, and you'll realize there that it is of course intercepted right at the point of optimum temperature. So here we move again. Enzymes work best at a specific temperature or around a specific temperature called the optimum temperature. For human, it's about 37 degrees Celsius. Interesting here, enzymes are denatured by high temperature. It's important to note that enzymes are not killed because enzymes are not organisms. They are actually chemical. You would have seen this in our first video on enzymes. So enzymes are denatured by high temperature. There we have the fire. So if you were supposed to go too close to too much fire for too long, then you understand that the enzymes will be denatured in you, and of course eventually you'll die. Here we have a leaf, and it is a leaf that is propagated asexually, but it has been exposed to hot water. There you're seeing it in the beaker with the hot water. Now the temperature here is way too high for this leaf. What is going to happen is that, of course, the enzymes in the leaf will be denatured, so of course this leaf will not be able to grow at the end of this experiment. And again, you're seeing the graph there showing that if, of course, we pass over a certain temperature, then the rate of reaction starts to fall. And of course, you know, eventually the organism will die. And we move on to the 7th property of enzymes. Here we go. The 7th property of enzymes is that enzymes are denatured by some poison, example cyanide and of course arsenic. And of course denatured won't work, you'll die. And here we have a skull that we got from our friends over there at Pixels. To access the skull, you'll find a link in the description below. I hope that you enjoyed the presentation. Please be reminded to like, share, and of course subscribe, and check out our many videos that are posted weekly. We also have 3 weekly live classes on science on a Wednesday, Friday, and of course Sundays at 5:05 p.m. Let's look at what's in store for us. How much do you remember? In today's lesson, we'll find out what are enzymes and how they work. Enzymes are molecules made of protein which speed up chemical reactions in the cell. Did you know that enzymes are not organisms? They are chemicals or molecules. Wow! Enzymes carry out the function without being changed. Here we have an enzyme. Enzymes have an active site which allow them to be specific in their action. Enzymes are also seen as biological catalysts that speed up the rate of chemical reaction in a cell. Enzymes are produced by the cell. Enzymes act on substances or molecules called substrate. Here we have an enzyme and the substrate. If enzymes are absent in a reaction, it would cause the reaction to take place too slowly, which would negatively impact the life of the organism. The enzyme reacts with the substrate in order to break the bond that the substrate has. Let's look at a reaction. Enzymes are responsible for chemical digestion of the food we eat. Starch is made up of many bonds of glucose. These bonds are broken by the enzyme amylase. They are reduced to maltose. Maltose is broken by maltase into a smaller version called glucose. Starch is now broken down at the level of glucose in a form in which the body can use it. Hence, glucose is absorbed by the body. Let's look at a animation. How the enzyme work. Here we have the substrate with bond to be broken. The enzyme with the active site, substrate will bond with the enzyme at the active site. Enzyme, substrate bonding with the enzyme to form enzyme substrate complex. The reaction continues. Substrate bond broken due to strain caused by enzyme forming enzyme product complex. Water is also a part of the reaction. The enzyme now releases the product and is free to combine with another substrate to continue the many reactions. If the yellow substrate here were to be starch, then the product here would have been glucose. Please see our video on the properties of enzyme. Thanks much for watching. Please be reminded to like, share, and subscribe. When you subscribe, remember to hit that notification bell so you can be informed whenever there is a new post. Mr. Wilson. Today we're going to be looking at symbiosis. Symbiosis refers to an interaction between organisms of different species. The interaction may bring benefit, harm, or no notable advantages nor disadvantages. Mutualism. Both organisms benefit from the interaction. The interaction between a ticks and a cattle would have been parasitic. However, the egret removing the ticks from the cattle would have formed a mutualistic relationship in which the egret would have benefited from getting food and the cattle comfort. The sea anemone and the clownfish, as is seen in the picture. Here, the clownfish would have gotten food and protection from the anemone, while the anemone would have gotten nutrient from the dropping of a fish. The fish would also remove organisms that would have, of course, harmed the anemone, which makes it a mutualistic relationship as both organisms are benefiting. Then there is also the nitrogen fixing bacteria that is located in the root nodules of leguminous crop. Here, the bacteria would have gotten somewhere to live while it is fixing nitrogen that the plant is able to use for its growth and other processes. Shark and the remora fish. This is, of course, another mutualistic relationship. Here, the remora fish would have actually hitched a ride on the shark, moving around freely, no energy used, while it would have been eating the scraps left over from the fish. Thus providing this clean environment and safe environment for the shark. This is a good example of mutualistic relationship. We're seeing the egret there on the horse, but the egret is popularly found among cattle, and the cattle are standing over there wondering, why is the egret over there and not over here? We have work to be done. We need some ticks to be removed. Predation. Predation is popularly called a predator prey relationship, and it refers to a relationship in which one organism, the predator,